everyone. Welcome to ROV Falkor. We are currently diving with ROV SuBastian on Cairns Seamount, on Herald Cays, off of Cairns, Queensland, Australia, in the Great Barrier Reef. And with us today, we have Dr. Dhugal Lindsay, who will be narrating and commenting on the midwater transects that we will be doing. And the principal investigator for this cruise is Dr. Brooke. And Brendan, I'll let you take it away and introduce Dhugal.
Yeah, thanks very much, John, and good morning, everyone joining us on this dive 376 of ROV SuBastian. So in this dive, we're descending down to about a depth of 700 meters on the northern flank of Herald Cays, for about 277 kilometers east of Cairns. During that descent, we'll be observing and collecting jellyfish, and we'll be joined for that part of the survey by Dr. Dhugal Lindsay in Japan. He's a jellyfish and plankton expert. Once we reach the bottom, we'll have a bit of a look around, and then start our ascent up the northern flank of Herald Cays. So now it's my pleasure to hand over to Dhugal. Dhugal?
Thank you, Brendan. Actually, on the screen right now, we have a large gelatinous planktonic organism. It looks like it's a salp, S-A-L-P, which is you know what a sea squirt is, one of those animals that attaches to rocks, and you poke it and it goes, and a little bit of salt water comes up and hits you in the face. It's a relative of one of those. It's a pelagic, floating, swimming sea squirt. And as it pulses there in the water, it's sucking in water through one opening, filtering it, and taking out all the good food, all these marine snow particles we see around, and ejecting the water out the back end in order to propel itself through the water. So it's basically a barrel of jelly with very large muscle bands to help it contract, and it sucks in and filters food. It's a filter feeder, can feed on very small particles. So even areas where the water is extremely blue, like the area we're in now, it looks like there's no plant plankton, phytoplankton around, but there's lots of little cyanobacteria and other small microplankton, and these salps can feed on that. So we'd expect to see quite a few of these pelagic tunicates swimming around. And we have our first one here at 137 meters. This is a solitary stage, and they come in two different generations, an asexual and a sexual stage, reproduced by cloning. So if there's a lot of food around, they can just keep budding and budding and budding to make very large chains of salp individuals. Depending on the genus of salp, above the species level, you have the genus level, which means they're very close relatives, like cousins. Depending on the genus of salp, Nakamedusa, that may be a solmissus or something similar. If we can take a look at this, these guys eat gelatinous animals. Almost the only thing they eat is gelatinous animals, like that salp that we just saw. So I think what we've seen here is a prey animal and then the predator swimming around there. They're very, very small. It's extremely difficult to follow one of these animals. It's floating around in three-dimensional space. The ROV is floating around in three-dimensional space, and especially at depths such as this, where we're quite shallow, so the length of the tether is not very long, which means you don't have a lot of freedom of movement with the ROV. You can be pulled back towards the ship just at that moment when you need to give thrust to chase the jellyfish. So very, very challenging to try and film some of these animals.
As well as the current, Dhugal?
As well as the current, yes. There's another salp, perhaps. See if we can get him. Maybe not. From a distance, because the animals are so transparent and small, it's often very hard to tell what they are until you get that sweet spot shot. That might have been a feeding filter. Oh, wait a minute. There's a solmissus down at the bottom of the screen. Let's go after him. There we go. There he is. Excellent. See if we can get a sensible count. A lot of midwater animals, when they're young, hang around in the surface layers where there's more food, and because they're small, they are hard to see by the predators. The larger you are, the easier you are to be seen, and therefore the higher the probability you'll be eaten. So animals tend to be small in the surface layers, and as they grow, gradually go deeper and deeper. One of the things we're trying to find out with this series of surveys is whether these small solmissus that are occurring in the surface are juveniles of the animals that are living deeper, or whether they're actually a different species. And we have a couple of samples and some tissue for DNA studies, so hopefully we'll be able to inform everybody of that in the future. No, that's good. All right. Definitely fewer than 20 tentacles. You got something in his stomach?
We're down by the lake. We're down by the lake.
Okay, let's keep on moving down. We are trying here to get a complete oblique transect, moving slowly forward and downwards as we go to characterize the different animals that are living here in the midwater above the reef. So as we're going down, all the marine snow particles we see around us, we're trying to make them hit the camera so we get at least one frame in focus as we go down through the water. How is the sound going here? Can you hear me more than the surrounding sound?
Yeah, Dougal, if you
or do we have a lot of interference?
If you could mute when you're not talking, that would be good.
Okay, roger. So this is amazing that we can be diving out here off the east coast of Australia, even though I am actually sitting here in Japan in the mountains. You can see all the green trees behind me.
So Dougal, Andrew here. Just wondering a little bit about the reproductive cycle of the solmissus, so that we were just looking at.
The group of jellyfish that includes solmissus has members where the juveniles parasitize other jellyfish, so they actually live inside the stomachs of other jellyfish and grow larger, and then are released as larger medusae. With Solomysis, we still don't really know. There haven't been good DNA barcoding studies. It's very hard to actually keep a deep water medusa alive and grow the animals out until you can tell what species they are as adults. So what we're trying to do is collect all the different morphotypes of the jellies that may do that, and then barcode the DNA. We're hoping to use the DNA barcode to then tell which species they are. I have a feeling Solomysis probably developed directly as a small larva floating around free in the water column, but I cannot prove that at present.
And clearly there's a range of different reproductive strategies among a lot of the jellyfish that you're interested in. Something just come on center screen there too.
Oh, this is interesting. Let's go into him. It looks to only have 6 tentacles, perhaps, and it looks to be a narcomedusa. Definitely want to have to take a look at this. It looks like it might have ingested a couple of salps in the stomach. Those 2 white dots could be food that it's eaten.
The ROV
Did you say it's got 8 tentacles there?
7 we've counted, yeah.
7, wow. If we can get a good in focus, from above like that, and one from the side, then I would definitely like to sample this animal if possible.
Our ROV pilots are working very hard to get a good image here, Dhugal. It's a lot of movement with the current.
That was a good one from the side. It looks like it might be a close cousin of Solomysis, Cunina perhaps. Still a little bit early to tell, but if we have the sample, we can look under the microscope and see if we can see lines of nematocysts, stinging cells on the outside of the bell down near where the tentacles are. He's an elusive little guy there. He's probably quite small. And the size of our ROV and the big powerful thrusters on it, it's very hard to try and keep this animal in the one spot, because the depth of focus is also not very great because of the lack of light reflecting back off the animal. So you're working with a very, very short distance where you can actually focus. Very, very difficult to do work on these animals.
Okay,
Okay,
Dougal.
let's try and tag and bag him if we can. Those white spots, it looked like one of them had just moved to the X umbrella. They could be parasitic amphipods, like little crustaceans that chomp on these jellyfish. It also looked like the tentacles were a bit shorter than I might expect. But it could be that the amphipods have been chomping on the tentacles. And if the Narcomedusa, this jellyfish, captures another jellyfish as prey, the amphipod will eat that. But if they don't, they eat the jellyfish itself.
We get it? So we think we got it, Dougal, but it was quite small.
All right, yeah, I imagine it's quite small. See if he's in the canister. It's very important for us to actually understand what is here, and unless we can actually tell what the animals are and what environmental preferences they have, we're unable to protect them and know what kind of policies are necessary for looking after the environment.
Dear, I think we've got him. He's tiny. Maybe a centimetre.
Okay, great. Yeah, okay, right, very small. That'll be interesting to know if those 8 tentacles, as they grow, sometimes tentacle number can increase as they grow, or it just could be a completely different species. We'll have to wait till we get him under the microscope back in the lab to check that out. Oh yeah, there he is, top right.
He's right at the top, Dougal.
Okay, great. Excellent.
Hey, onwards and downwards.
Okay, just a second. I'll be back in 10 seconds.
It's his, yeah, very colourful background from Dougal.
Okay, I'm back now.
The other guy is streaming live, so not to say anything with personal information.
There we have something, set a screen. Looks like a mucus filter, probably discarded from a pteropod feeding web. Pteropods are little pelagic snails. Their foots have evolved, like the foot of a snail has evolved into 2 wings. So they flap around, floating around, swimming around in the ocean. and they excrete these mucus feeding webs, which they use to catch small phytoplankton, kind of similar to the way a kelp catches small phytoplankton with a feeding web. And then they ingest the mucus along with all the things that's stuck to it in order to feed. Looks like it might be another kelp, solitary individual. Yes, it's a kelp. The white ribbon-like structure you see at the top of it there is a chain of little baby kelps that are all joined together. As they grow out, they get bigger and bigger the closer they are to the end of the chain, and these animals will detach from the mother and swim around as a very long snake-like chain of kelps, filtering all the food out of any water that they pass through. One of the things we're doing with these surveys here is as we're recording the video, we have an instrument called a CTD, which is a conductivity, temperature and depth meter, which has a dissolved oxygen probe attached to it. So we're able to look at the environment these animals are in to see what is the temperature, or what is the salinity, what's the oxygen content, the water density, etc., where these animals are living. And because we have been collecting data sets like this from places like off Japan or the South Pacific or Monterey Bay in the United States, we can piece together the habitat preferences for all of these animals. And it's important to get this kind of data because if we find keystone species that are very important to the ecosystem as a whole, and we can tell what are the limits of temperature that they prefer to inhabit, we can see what the effects of global warming or climate change may be on the distributions of these animals. And if the animals are no longer able to live at temperatures where their food is, for example, then we have big problems for them, especially if they're one of the keystone supporting species in that ecosystem.
Good morning, everyone joining us remotely. We're currently undertaking a dive, ROV SuBastian, diving off the northern margin of Herald Cays, 277 kilometres east of Cairns. We're descending to a depth around 640 metres, and on the way down, acquiring samples in the water column, primarily jellyfish. As we reach the bottom, we'll have a little bit of a look there, and then we'll be taking our ascent back up margin of OK. And during that phase of the survey, we'll be acquiring high-resolution imagery, as well as bed geology and benthic organisms. During this descent, though, we're very lucky to be joined by Dr. Dhugal Lindsay, just via satellite from Japan. And jellyfish, and Dhugal's providing us with some expert commentary as we dive down through the depths. Okay,
Thanks, Brendan. I'm sitting here in Japan, many thousands of kilometers away from where all the action is happening, looking in real time at a depth of 180 meters off the coast of Australia. Very exciting world we live in. Unfortunately, I was unable to personally take part physically in this cruise due to coronavirus travel restrictions, but our team has really excelled itself in putting all of this together so that we can still make this survey happen the way it was intended. Very thankful for the opportunity to be taking part in this history-making expedition off the east coast of Australia. Doing this survey here on my notebook computer, sitting out here in the forest. One of the challenges I have here is, without looking at a nice television monitor, which I'd normally be looking at during one of these surveys, is all my fingerprints on the screen look like they're jellyfish to me. So I have to, oh, there we go, there's one, there's a real one. Possibly a ctenophore. If we can see the reflectance of the comb rose there, which will let us know if it's a comb jellyfish or something else. Certainly looks to be a comb jellyfish. Comb jellyfishes are actually not very close cousins of regular jellyfish that have stinging cells. They're in a completely different phylum. They're more different than goldfish and people, for example. So although they're transparent and soft, that's pretty much where the similarities end. These animals have 8 rows of macrocilia, very large rod-like proteinaceous structures, which are put together like kind of like the fingers of my hand. There's many, many small fibers which are fused together, and as I'm moving my hand, the ctenophore beats its comb rows, and it uses them like oars or paddles to propel itself through the water. Ah. It could be Thalassocalus. I think we're looking side on at the animal now. Yes, Thalassocalus. So this is a medusoid ctenophore. It looks like the same shape as a jellyfish. Oh, wait a minute. No. It's some other kind of lobate. Huh. I can make out the stomach, the bright white line in the center there. Looks to be the stomach, and on either side, I think they may be auricles, ear-shaped projections covered in cilia, which the animal uses to scare its prey. So the prey will flip away from the auricles and get attached to the lobes, where they can be moved towards the mouth for ingestion. It also seems to be quite a small animal. Since I'm seeing this video as it's been sent over the internet to here in Japan, the actual quality with which we're recording this video is much greater. So because we're recording at a high quality, I'll be able to take this video back in the lab and hopefully get proper species identification for some of these animals that, you know, they're a bit laggy coming here over the internet to my eyes right now. It looks to be a young lobate ctenoform rather than thalassocalus. Belongs to the order Lobata. You can see the great big fleshy projections now on the right-hand side of the animal. So hopefully we'll have enough video here that I'll be able to ID it back in the lab. Oh, that's nice. When we see these images recorded at the original resolution.
You're happy
Okay,
to
let's
keep going?
keep heading down.
Yep, great.
Yep. Either a kelp chain or Bugmania siphonophore down here. Ooh. That's a kettignate in the foreground out of focus. What's that at the back there? Ooh, could that be? Looks to be the tail of a siphonophore. May or may not have the actual swimming gills attached. It looks like it's a ghost tail, so we can keep moving. Brendan, are we very close to a reef which is actually above the surface of the water here, or is the reef way near fully submerged?
Yeah, that's correct, Dougal. There are surface breaking reefs in this area, unlike at Cairns Seamount, where we were the previous 2 days. There's a number of reefs and cays in this region.
I'm very surprised by the amount of organic matter, both animals and marine snow, where we can't really tell what it is. I think perhaps there might be a very large input from the mucus excreted by corals as they're either exposed to the sun or to wave action or something like that, and possibly all these masses of mucus being transported from the reef out to the open ocean here where we're diving is putting a big input of food that allows all of these animals to live here. What would you say to that hypothesis?
Yeah, it seems consistent with what we know about this area and what we can see from up on deck. First time for several days that we've actually been able to see any land mass. And yeah, Herald, the large part of a large cay within this Herald Cay network is visible from the ridge. Very beautiful looking island. So we're now at 201 metres below the surface on our descent on the northern margin of Herald Cays down to a depth of about 640 metres. ROV SuBastian dive 376, located about 277 kilometres east of Cairns. First leg of our Seamounts, Canyons and Reefs Coral Sea research cruise with the Schmidt Ocean Institute.
It's very nice the way they're driving the ROV so that the particles are coming from below and in front of us. Hopefully as they come towards the camera, we'll get at least one frame where every particle is in focus so we can identify what these different particles are. The term we use for particles such as this is marine snow, basically because it looks like a nighttime snowstorm when we're diving down here. But they can be all manner of different particles. Some of them may be inorganic, but many of them are organic matter, which provides food to the organisms that live below, or the organisms that are floating or swimming through the water column. So a lot of these particles that just look like white spots on your screen here are probably things like fecal pellets, the poos from other jellyfish and salps or other animals. That there was a discarded filter from a lavation. There's mucus that's probably been excreted from corals or from jellyfish floating around here too. Oh, that looked like a siphonophore. The molts of crustaceans, as they grow, they throw away their exoskeletons. And these, the scales of fish, all of these things are floating or sinking through the water here and providing food as a rain of marine snow on the benthos, these seafloor living animals that are lurking below us. A lot of animals hang out and wait for the marine snow to float past or sink past, and they eat the snow. There are other animals that migrate actively to the surface at nighttime. This is another lavation. That white spot in the middle of that mucoidal particle there is the body, the main body of the larval-looking tunicate. And the semi-transparent structure around it is its feeding house. It beats its tail in the water to create a current, and this draws water onto the filter where it traps the little particles it beats, and then it ingests these particles and eats them. As it's continually making this current to bring particles into the filter, the filter tends to get clogged after a while with particles that are too big to fit in the lavation's mouth, that clog the feeding house. Oh, what do we have here? Is this a Calicofton siphonophore? Very small animal, whatever it is. Here we go. Can you make out if it has a bell there, that white spot? There's also the possibility that it's a rhizarian. Yeah, it's looking more and more to me like a single-celled planktonic organism called a rhizarian, like a radiolarian or something like that, which maybe has excreted a long string of mucus. I don't know if anyone who has a slightly better video feed than me can see like glass spines coming out from that central white mass.
We're just trying to get a closer zoom, Google.
Roger that.
A bit like the world's most difficult computer game, Dougal. Get a good image of this.
Yes. It could be a larvation that just doesn't want to throw away its inner house. If it is a larvation, there won't be any spines. It'll be an oval-like white mass, which is the body proper, and then a transparent tadpole's tail, which will be half embedded in that mucoidal structure that's surrounding it. It does look to be bilobed, so I think perhaps that's an oikoplurid that just doesn't want to let go of its filter house. It must be very hungry, and it has an investment in that filter that it doesn't want to let go. Yeah, I'd say that's what it is. It's an oikoplurid, a labation. Thank you for that. That was pretty challenging.
Shall we proceed?
Yes, please. Let's keep heading down.
We're now at a depth of 217 metres on our way down to the seabed, which lies at about a depth of 640 metres. During this descent, we're observing and potentially sampling some of the jellyfish and planktonic animals we can see in the water column. We're on the seabed, we'll have a bit of a look around and then begin our ascent back up this northern margin of Herald Cays. Herald Cays sits on a relatively shallow part of the Coral Sea called the Queensland Plateau, and of course this is located within the Coral Sea Marine Park. We're fortunate on this descent to have Dr Dhugal Lindsay join us live from Japan via the ROV Falkor's flight communication system. Dhugal is an expert in plankton and in particular jellyfish. Very much enjoying being able to join us from forests of Japan.
Thank you for that introduction, Brendan. Yes, I do a lot of work on jellyfish now. I actually started in marine biology working on fish and shrimps that we caught in nets. We drag a net through the water column that we're actually looking at now with, through the cameras. And most of what we catch is crustaceans or fish. There are very, very few jellyfish in the net samples I collected back when I was a student and a young researcher. I did my first dive in a manned submersible back in the late 1990s, and everything I saw was a jellyfish, it seemed. So it completely changed my outlook on what are the main inhabitants of this mid-water dark twilight zone. And it's from there that I started to get interested in jellyfish and how important they are to the ocean's ecosystem as a whole. These animals, they're both very important predators that feed on anything else that's living here, floating and swimming in the water. But they're also good food for a lot of animals. It used to be thought that jellyfish, being mostly water, did not provide enough nutrition to predators. And so predators would not eat jellyfish, they'd go off and eat something else instead. But we've found more and more now that a lot of different animals are eating jellyfish. And recent advances in DNA barcoding, where we can find a barcode of DNA that will identify an animal, a certain sequence. And we're trying to collect DNA barcodes for each of the species that's living here, so we're able to identify them in the guts of fish or shrimps or other animals that are actually eating these jellyfish. Because the jellyfish are mostly composed of water. Up to 98% of their body mass, wet weight, can be water in some of these species. They get digested very rapidly. So when you cut open the stomach of a fish, it's very, very rarely that you find anything that looks remotely like a jellyfish inside. So the way we're getting a handle on this is to go out with something like an ROV like this, collect the jellyfish, see exactly what they are taxonomically, morphologically, and take a bit of tissue, sequence it to get a DNA barcode, and then these barcodes can be used by probing the stomach contents of the fish or the turtles or the shrimps or whatever it is you're looking at to see whether that has been eating this species. We've been able to apply this technique to things like Antarctic seabirds to see that jellyfish are actually an important part of the diet of some seabirds. It looks like they land on the beach and peck away at jellyfish that have become stranded there, and this tides them over when they can't get enough fish to eat, for example. And in an area like this, wow, what do we have here? Oh, I don't think I've ever seen anything that looks like this. It looks like it might just be a strand of mucus, maybe a dead animal trailing something behind it. It looks... Yes, it's definitely not alive. It's a mucus ejection from something. And I don't think we'll be able to identify exactly what. Although it does look like a heteropod mollusk, perhaps. These are gelatinous mollusks, relatives of the shellfish. Thank you for that. That's in focus. I should be able to tell what it is later. Let's keep going down to make sure we get a complete transect of the water column. These heteropod mollusks prey on things like salps, which we've been seeing earlier on in the dive. So they're gelatinous predators which feed on gelatinous prey. So not only do the gelatinous prey get digested in the stomachs of any predators, so you can't see what they've been eating, but the predator itself being gelatinous, if you try and catch it with a net, they're often broken up into little pieces, kind of like straining them through the mesh of the net and having transparent spaghetti coming out the other side. So it's only with tools like the Schmidt ROV here on the Falkor where we're able to actually see these animals in their environment and to get a handle on how important they are here, like what percentage of the animals that are living there are which different group. And if we're really lucky, we can actually come across one feeding, or the jellyfish being transparent, we may even be able to look into their stomachs and see what they've eaten. Glad people can't see into my stomach and see what I ate for breakfast. But it's very useful for looking at the ecology of these animals while they're still alive down here in the deep sea. And it's streaming to you directly here from the east coast of Australia, 260 meters depth. There really is a lot of mucus here. I haven't seen anything that looks like a Bathycordius house, Bathycordius being one of those large lavations we saw during the survey yesterday. More mucus. So I'm really wondering if it's laterally transported mucus from those corals. I think it might be a good idea for a subsequent cruise, now that we know that there's a lot of these large mucus particles out here, to get something like a detritus sampler to actually sample one of these big pieces of mucus and try and do DNA barcoding on it. And because as the animals excrete the mucus, their DNA should also be trapped in that mucus, and we may even be able to tell where the mucus is from. It may not be very sexy science. It's not like we're looking at a giant squid, but it's very important to understand how these ecosystems function. Perhaps in some subsequent expedition we might do in a few more years' time. The data we're collecting on this cruise will keep us busy for a very long time after this cruise. We'll be going back through these videos frame by frame, trying to count the numbers of different animals and what kinds they are, and recording, documenting exactly what was here, and then trying to interpret by looking at the CTD data and other information why the ecosystem is put together in that way in this place. And then by comparing it to all the other survey sites we have, try and get a handle on the processes that are involved to make the structure be that way. That's a Physinect siphonophore, I think. It's a siphonophore at least, which is a colony of jellyfish polyps that have taken on a pelagic existence. Rather than attaching to a piece of wood or a rock or the sea floor, they attach to each other. And some of them have evolved to have jellyfish-like swimming bells. So they're able to propel the animal through the water, dragging the rest of the polyp colony behind it. The different polyps all have different functions. Some polyps have developed to become gonadendrons, for example. Others are bracts, which protect the gonads from predation by other animals. Some of the zooids take the form of stomachs with an attached tentacle that they use to prey. And there's a whole range of different zooids. There are polyps that have developed to taste or to feel, dactylozoids, looking kind of like the fingers on my hand, long, thin zooids. And all of these are attached together, swimming or floating around here in the mid-water, continually eating their prey. One species of siphonophore can reach lengths of 40 meters, which is twice the length of an adult blue whale. And these guys hang around at depths quite like this, actually, off Japan, maybe 250 to 350 meters depth. They often hang around above the schools of krill that migrate to the surface every night to eat. And as the krill migrate upwards towards the surface to get to their food, the siphonophores hanging down their tentacles, 40 meters of tentacles, hundreds and hundreds of stomachs, sitting there and lying there in wait. As the krill pass through, they eat them all up. The krill eat in the surface waters, and as they come back down again, they get eaten up again. That's a solitary salp we see there. Top right of the screen, and in the middle, this looks like possibly a larvation. Yes, a larvation house. Oh wow, it's got things embedded in it. Oh, yeah, if we can zoom in and see, are they crustaceans or something? Feeding on the house, yeah. It's a discarded larvation house. The filter has become clogged with lots of food particles. Can no longer filter the smaller particles that fit in the larvation's mouth effectively. But these are like packets of food that sink quite rapidly down into deeper depths. So many planktonic animals will be swimming around in like a search pattern. And then as the particles sink down, they leave a trail, a smell of food. And so the crustaceans or other animals will be swimming in this search pattern and come across this trail, and then they'll swim rapidly down to find that particle and then chomp on it. So it really is a very three-dimensionally structured environment with sinking particles, all leaving trails of smell. Sometimes pheromones. The animals will be releasing hormones as they sink, and the males will go in after them, or particles that smell good as food will also be leaving trails. So these animals, as they're floating or swimming around, are looking for these trails to try and find what they need, either reproduction or sustenance.
Center
Ooh,
screen.
squid? Is that a squid? What is it? Holy mackerel. Crustacean?
Like it's
No,
lost
it's a
all its
jellyfish.
jelly, yeah?
Oh, it's a Bathychorus. Wow, that's weird. Is it missing the bottom half of the bell? Or is that the actual structure of it? Huh. That is so weird. Oh no. No, it's a crustacean. It looks like the larval form of something with like the legs splayed out to the sides. This may not be a jellyfish, but I think it could be scientifically very interesting. Perhaps we should sample this. I'm sure we can find someone who will be able to tell us what it is. And it's not very often you come across larvae to know like where these animals spend the very important larval stages of their life cycles.
You'd like to sample it, Dougal?
Yes, I think we should. I've been diving for
Yeah, sorry.
over 20 years and I've never seen anything like this.
Sorry, Dougal, we're not able to sample that one.
Okay. Just some close-in video then, if possible. Wow. The angle on the body suggests a pasiphaid shrimp, but I've never seen them hold the legs like that. I assume the legs are out like that to make the sinking speed as slow as possible. Oh, wow, that's nice. There's all that feathering on the legs also to increase water resistance. Yeah, let's hope we got that in the stereo cameras as well to get a size on it. Thank you. Okay, I guess we'll keep going down. Try and identify that from the images later. So, a great thing about ROVs is you actually have a collection of images that you can send to other researchers who may know more about the animals than you. It's always a problem with research cruises. There's only a limited number of beds. And because there's such a high diversity of animals that live out here in the deep sea, you can't always get all of the specialists you would want on the ship at the same time. So the kind of technology that the Schmidt Ocean Institute is championing here with live video streaming, being able to get a variety of other experts who may know what these animals are, being able to see the animals and provide advice is really going to change the way these kinds of surveys are done going forward. That's nice. I'd put my money on that being a pasiphaid shrimp larva. Very cool. Notice the way the antennae are out curving around to either side of the body. That's probably to sense any predators. As soon as they touch one of those antennae, this shrimp will flip backwards to try and escape. Okay, that's really good. I think we're fine.
Dougal,
If that was a passive fade.
Dougal, it
Yes.
seems we can sample that if you really want it.
Is he still there?
Is there?
Yeah, if we could try, that would be good. We've got a lot of nice images there of from the front and from the side, etc. One of the other things we've been trying to work on is making an image database to train artificial intelligence to help us to analyze videos such as this. So if we can get a positive ID, every different frame of every different angle of image we have of this animal can be used to train the artificial intelligence to recognize it next time. That way, even if the specialist is not on board here looking at the video, the computer will be able to say, I think it's this, and give you kind of an idea as to what it might be. As it senses the water flowing into the suction sample, it will probably give quite a vigorous flick and try and get out of the way. So you have to be ready to follow it. Oh, close. It's challenging in a different way to the jellyfish. Ooh, there he goes. Oh, you got him. Keep sucking, keep sucking. He might be holding himself, trying not to be sucked down that tube there at the entrance. I've had a squid like hold on like for dear life. Oh no, looks like you got him. Well done. Yes. Very expertly done.
Thanks to J-Rob and Cody.
Wow. It's one thing that's so exciting about these dives into the deep sea is you see something new every time.
So Dougal, what's the largest siphonophore or the largest you've seen in all your at deep sea?
Me personally, I've seen one where we weren't able to follow it all the way to the end. We kind of had it in our cameras and went from the head, so to speak, the swimming bells, and we're like moving along trying to count the stomachs and what proportion of them had prey in them, because you can actually see like a stomach looks shrimp shaped because the stomachs are so soft. They take on the shape of whatever the animal is that they've eaten. So we're trying to do a survey to see how many of these stomachs had food in them and what it's eating. We got to like stomach number 40 something and lost the animal. It just kind of swam or floated away. Oh, here's a siphonophore. Possibly a Bugmania. It's a Physoneck. It's got a float and on the top. Seems a lot smaller than the siphonophore we were just talking about. 1, 2, 3, 4, 5, 6. Over 6 stomachs. Very widely spaced. Ooh, could it be a Roticosia? We're not yet in Antarctic intermediate water depth, so I wouldn't think it likely, but it certainly morphologically looks like it could be. There he is. It's very hard to tell these siphonophores apart using video alone, because as the animals were originally described, they're normally described from samples caught in nets. And you can imagine dragging a plankton net through the water and having one of these polyp colonies, a siphonophore. Hundreds and hundreds of different zooids make up this siphonophore. And you may have multiple individuals and even multiple species caught in the same plankton net. This looks like a... Oh, that's a Calicophron. This is possibly Rosacea. If you can take a look at the nectaphores, the large gelatinous mass at the top of that. This is a relative of the guy that can go to 40 meters in length. They can be very, very top-level predators. Like a shark can only eat one thing at a time, but these siphonophores can eat like hundreds and hundreds of things at a time if they had that much prey around.
Smorgasbord.
Yeah. And so when you catch them in a plankton net, the original descriptions sometimes had, one part from one animal and one part from a different species, and they described them as the same species. So you've got to kind of put things together as a jigsaw puzzle to see exactly what they are. This is good, this is good. So it's a praeid, not prayer, but a praeid. And if we can zoom in on that bell, we'll be able to take a look at the somatocyst, which is where all of the food, energy reserves are stored. And the shape of the somatocyst is what lets me tell what genus it belongs to. Some of them have these bright white somatocysts going back into the bell. Some run along the surface of the bell. Some branch out and we've got branching pattern. And we have to see those kinds of things to be able to tell what genus or species they are. You get a pretty good guess on what it is, but already it looks like a Rosacea, just the form of the nectaphores, mostly cylindrical. It's 10 or 12 stomachs. Hopefully I'll have that video in 4K at high quality to look at later, so hopefully I'll be able to get an ID on that animal.
Yeah, so we better
going down.
Yeah, we better get going, Dougal,
Yep.
to the bottom in time to
Would you like me to close my eyes for a while? I can put a blindfold on for you. still very amazed at the number of animals there are here, given that you wouldn't expect the surface productivity to be very great in an oceanography kind of sense. So it must be the proximity to the reef. It's also quite diverse. I think it's more diverse than the Cairns Seamount area we were looking at before. That was mostly salps and tunicates. Here we've got all of the major groups. Narca Medusa. Looks very small. Possibly a Solmithus. Certainly looks like a Solmithus. 16 meters. Got his tentacles held out in front of him. Oh, and there's a salp. That's what they prey on. So they hold, and it looks like there's a salp in the stomach as well. You can see the that bright white spot. Could be the stomach of the salp. Could also be a parasite that's living in there. That's some nice videos. Once we get a couple of in-focus shots from above and the side, that's enough to tell what it is, and we can keep on heading down. Oh, that's nice. Oh yes, and it's got stomach pouches. Nice big oval ones. Very nice.
Happy with that, Dougal?
Yep. Yes, thank you. Just keep going down, get you to your seafloor. Very happy with the dive so far. If you are having time constraints to get a good benthic transit, I'm okay with you hightailing if you need to. Siphonophore. Wow, there are almost no siphonophores over the Cairns Seamount, and they're everywhere here. Goes to show how you cannot just pick a point at random and say this is representative of the entire ecosystem, the larger ecosystem. It's really important to get this fine-grained, fine-scale data from different points. Obviously in the benthos it's even more so, but even for the pelagic, the plankton, where they're all floating and you think they'd be like mixing up and being relatively homogeneous, we can see it's definitely not the case.
They're quite different settings as well, Dougal. The Cairns Seamount was in that, 1,200–1,300 metres of water in the Queensland Trough, and now we're up on this plateau maximum depth. Area we're in now is about 700 metres.
Roger that. I feel like I'm jumping into hyperspace here. Ooh. Is that another siphonophore? How far above the bottom are we now, Brendan?
Yeah, we're a bit, yeah, over 150 metres.
Roger that. Is the slope here very steep?
It is as you come up closer to the northern margin of Herald Cay. It's especially steep. There's a bit of a ledge we'll be going up. That's at about 600 metres, and then it flattens out a little more, and then it's quite steep from 400 metres up to 100 metres.
So there could conceivably be lateral transport of matter from like all sorts of different depths out here.
Yes, I think so.
Just seems that most of the depths we've gone through have had lots of animals and lots of particles around. Normally way out in the open ocean, the top 250 meters, 300 meters is really bare. Virtually no animals living there that are large enough for us to see with the cameras. Whereas here we've just had animals the whole way down.
Well, it's quite a unique setting here, having this plateau with so many huge reefs sitting on it. These form reefs that we're surveying the margins of are quite unique to have so many of them in relatively.
Ah. That's a Physonex siphonophore. What is that? A Gamma Okini or possibly an Arena? We get close enough. Looks like it might be a Galma Okini. Agalma Okenii. Oh, it's orange. Yeah, very nice. So in this siphonophore, very different to the last, the cyphosome, which is the part of the animal that has all the stomachs attached, is very rigid, and the bracts are locked together, kind of like a Rubik's Cube or Jenga blocks or something, and the tentacles are coming out from the cavities between the bracts. And so these bracts are like making this three-dimensional matrix that is protecting the zooids inside, the reproductive zooids and the gastrozooids from being eaten. There are some animals like charistid fish, where the juveniles hang around these siphonophores, and when the siphonophore catches the prey for them, they just eat the stomach containing the prey. And so they just hang around these siphonophores waiting for them to catch food for them, and then pick off the stomachs as they become full. This guy, the Brax, will be protecting it from other animals trying to do things like that. Very nice. Hopefully we'll get some size info on that as well with the stereo camera. So definitely the diversity here is higher than it was on the Cairns Seamount. Is it possible to make the camera feed just a little brighter, please? That's squid?
Looks like
Yes,
it.
it's squid. Oh, wow. You can even see the bioluminescent organs. Wow, nice. We still there. Is it a fire squid? Wow.
Right away.
Yeah. Oh, they're fast guys. Very nice.
Beautiful.
Those big eyes. Oh, that's good, yeah. Oh, yes. Are they the photophores? Those blackish looking dots on the surface? Oh, I have to show this to Mike Vecchione at the Smithsonian. He'll know what it is. Oh, beautiful. So a lot of the animals that we think of as surface livers, like tuna, actually make forays down here into the mid-water to come and feed on squids such as this, squids and other fish. Because in the surface waters during the day, there's not all that much happening away from the reefs. And most of the animals have come down here to hide. So they'll come down to dive down really deep to snack on things like that squid.
We've got about another 250 meters to get to the end of our dive before we reach the seabed. it'll be the end of this mid-water transect where we've been looking at the plankton and jellyfish. We'll get to the seabed, have a look around there, and then we'll start our dive back up the margin of Herald Cays. And on the way up, we'll not just image the seabed, but hopefully we'll be able to buy some samples of geological features as well as anthos that we find, in particular looking at deep water corals.
Theropod.
Joining us today from Japan is Dr Dhugal Lindsay, an expert in plankton and jellyfish. You've been enjoying the dive so far, Dhugal? Yep.
Oh yes, you bet I am. It's been great seeing all of these animals down there, thousands of kilometers from where I am now. Actually seeing them alive, swimming around, and looking at their behavior as well. Very exciting stuff. We just had I think we have a fish on the screen here. It's moving very fast for something living this deep. We had a theropod mollusk, which is a kind of a snail, a swimming, floating snail, which just went past the cameras a minute or two ago, with a triangular looking shell, maybe clear. These animals have shells made of calcium carbonate, which means that as the ocean acidifies, the shells are likely to become thinner and dissolve, and we don't know what's going to happen to them when things like that happen. A lot of these animals, oh, what do we have here? Something center right. A lot of these animals are very, very small and mostly transparent. That looks like a narcomedusa. There it is. And very, very fragile. Very difficult to sample them with anything other than an ROV. Oh, not an narcomedusa. Maybe a trachea. We're taking video now that we'll be able to take back to the video labs at our respective institutions to analyze properly. And getting an ID on one of these animals normally involves jogging through the video frame by frame, finding the different bits that are in focus at any point, and looking in multiple books and scientific papers, some of them published in a variety of different languages over the last century and a half, trying to tell what these animals are. Can take more than an hour to identify an animal in some cases, if it's not one you're very familiar with. Oh, was that a ctenophore, perhaps? Some animals are very easy to recognize in the video because they have the macro morphology is different. So even just with images, you can tell it's this or it's that. Some of the animals, the differences are much more subtle. So you really need to have a good quality video to tell them apart, or you may need to sample them and look at them under a microscope. It looks like a ctenophore. You can see the 8 comb rows at the bottom of the animal there facing down, and the large lobes towards the top. Spinning around now, we have a side-on view. Quite soft gelatinous material. Should be a rounded Balanopsis type of a body to it. You can see where those tentacle bulbs are. You'll have a better idea. These ctenophores, there's a large number of undescribed species. The percentage of undescribed species may be somewhat similar to the percentage of described species. This is because they're so fragile that it's very hard to get an animal in good condition back to the surface to look at it in a lab. And for most taxonomy, you need to have more than one individual of every species, so you can be sure that different morphological differences are not the result of a mutation or something like that, but it's actually a species level difference. So it can be very hard to describe some of these ctenophores. Thank you for that. It's nice when they move around, so you get angles, like a variety of different angles there to allow you to see all of the morphology, the shapes of the different structures inside the animal.
Hello.
A lot of these little particles of marine snow are probably very small crustaceans called copepods. There may be some ostracods in there as well, which is a crustacean that looks like a bivalve, a pippy, or a clam, where the shrimp is inside. That's a larvation house. A weird looking one, but yes, a larvation house. So not all of this material will be dead detrital matter that's sinking down, but some of it will be live animals. There's an Archimedusa, a Solmissus perhaps. Looks like a Solmissus, 533 meters depth. White socks. The distal ends of the tentacles are white. Oh yes, that's a different species to the one we saw just before in the surface layers. If we're able to, once we get some video of this, I'd like to suck him up, please.
And which species is this to?
Solmissus, S-O-L. Oh, it's got gonad tissue. Look at those whites, the white spots on the edges of the stomach pouches there.
Roger that.
Oh yes.
Green grabs of all of this.
So it's a fully mature Solmissus individual. It looks like it's ingested something in its stomach as well, so we could get some information on what it's eating. The gonads tissue there on the edges of the stomach pouches are all there, and it looks different to the guy we find off Japan. The species name this would normally be given now is Solmissus incisa, I-N-C-I-S-A, but we believe this is a species complex of cryptic species, and we're trying to tease apart exactly which species is what and how to delineate the different species based on morphology, not just on their genetic sequences.
And the white socks on this particular species is
it may be definitive, but I can't yet be definitive about that.
Be right to Sam Dougal?
Yes, please. It looks like there's higher concentrations of nematocysts, or stinging cells, on the distal halves of the tentacles, which would make sense because when we came across this animal just before, all of the tentacles were pointing upwards. So it's the distal ends of the tentacles that would encounter the prey first. Therefore, that's where the stinging cells should be. Some of the species look to deploy their tentacles, half of them up and the other half of the number down. So there seems to be some behavioral differences that we might be able to link to morphology as well. Oh, this is a great animal.
There you go.
Got him.
He's in. Awesome. He's in the canister too. Excellent.
bit easier than the planktonic shrimp.
Shrimp, yes. Yeah. Some jellies are very fragile, so they're hard to deal with like that, but they don't swim as fast as some of the other critters, so that's a nice thing. Awesome.
Okay, we've got about another 100 metres, 110 metres before we reach the seabed and switch over from our midwater descent survey to a survey back up northern margin of Herald Cay.
That's the altimeter data seems to have is coming through now. A pinging sound off the bottom to tell how far above the bottom we are. As we come towards the seafloor, normally one would expect the numbers of animals to increase as there's more food associated with the seafloor than there is with the water in between. However, this survey today, it looks like there's a lot of lateral transport from the nearby reef. And no matter what depth we're looking at, there's oodles of food and oodles of animals. Very exciting place to be diving. It's quite interesting that there's very few animals with pigmentation. The Siphonophore agalmo-akini we saw before had reddish stomachs. That's probably because it eats bioluminescent prey, and the prey that it ingests give out blue light, and the red pigmentation of the stomachs helps to act as a shutter to stop this blue light from being visible from the outside of the animal. Because if your stomach is glowing, then you're visible to things that might want to eat you. So in some cases, they'll have pigmentation because of this, but because of the clarity of the water, probably most of them, you would imagine, would be transparent, which does seem to be the case. That's a salp chain. These are all clones budded off from the solitary salp. Looks to be the genus Salpa. All swimming along there in a chain. Those white spots you see there are the stomachs. They ingest water, filter out all the food, and then eject the clean water out of the back half of the animal. And swim through the ocean, eating as they swim. These salps produce very large fecal pellets. So they package tiny little phytoplankton, eat them, mostly digest them, but there's still a lot of good food contained in them. And these fecal pellets will sink down to the deep sea quite rapidly to provide food to the benthos. Oh, wow, ctenophore. That's a sediped. Oh, a metenzid, I think. Filiform tentacles, tentacle bulbs, 90 degree to the axis of the animal itself. Yeah, this is an undescribed species of sediped ctenophore, which looks similar to one we filmed off the coral reef in Okinawa, in Japan. Be very nice to get shots from all angles of this. What we need to be able to see is the canals that are branching off the stomach out towards the comb rows in order to get a species ID on this.
What is that?
Yep, I think we might have got that shot there. See the canals? Now 60 meters above the sea floor. Still lots of animals. Krill. The whale food just swam past. That looks to be an amphipod, another crustacean. They often occur in larger numbers closer to the seafloor. So it looks like he's up swimming around looking for food up here rather than on the seafloor right now. There's a salp chain, again, the genus Salpa, asexual clones. Those salps also undergo diel vertical migration, swimming around in the surface waters at nighttime, filtering out all the phytoplankton, and then coming down to depth during the day. If those salps occur close to the seafloor, they can be grabbed by a crab or caught by some coral and eaten by there, providing valuable food to the benthic community.
So it looks like the end of our descent is in sight. We can just make out the seabed.
Thank you for that very interesting mid-water transit.
You're welcome, Dhugal. Thanks for the commentary and yeah, all those amazing insights into the world of jellyfish and plankton. We'll sit Sebastian down on the seabed and have a bit of a look around.
Okay, I'll help try and conserve your bandwidth so you get some benthic people able to give you some comments. So I think I'll sign off now and looking forward to hearing about what you see on these benthic transits later on today.
Thanks very much for joining, Dhugal. That's Dr. Dhugal Lindsay, who's been joining us live from Japan. So we made our descent down to 142 meters to the seabed, margin of Herald Cays, Coral Sea Marine Park. So we're now on the sandy seabed. We're just preparing to collect a core of sediment at this site. And you can also see very pretty soft coral, Jeremy. So at the moment the ROV pilots are just preparing ROV to collect a few samples, and then we'll be making our way back up the northern margin of Herald Cays. So the main objective today is to acquire the very high resolution video and still imagery that we do using SuBastian as we go back up the margin of the cay. And as we go along, we're also able to quite strategically sample the bed features, rocks and sediments. So very importantly, pick samples from the different biological communities that are growing on the seabed. Just about to get underway with SuBastian. We're changing over the ROV pilots and we'll be very shortly underway. You're watching live video feed from ROV SuBastian. SuBastian is on seabed, okay, at 638 meters, but a temperature is 6.6 degrees Celsius. Just reached the seabed after our dive through the water column, and we're preparing to collect a sediment sample and also way up, firing high resolution images and collecting samples of the geology and biology as we move up the margin of Herald Cay. It's all about looking at the relationship between the geology of the margins of these reefs and the distribution of different biological communities, as well as linking that to the water column and magic organisms, whereas we descended down to this point, and provide some samples of the tectonic life forms. So Chris and Adam, the ROV pilots, are now just about ready to collecting a core. Yes, we've got a turtle having a free ride on Sebastian's box. So hopefully Isla can see that turtle there by her dad Dean. Yeah, we had a very special, very deep water turtle come down with us today. And Sebastian is just about ready to collect a core for us. Robotic arm grabbing the short core for her. Yeah, unfortunately that core barrel, the collar for the core holder's caught up, so we're going to select an alternate barrel, I think. Like we're going to have another go. The sediment's very, very loose, and it was very hard for the seal at the top of the core barrel to create enough of a vacuum to keep that sediment in. we seem to have managed to have did a good sample there. It's very unconsolidated and watery sediment, so a bit of a challenge getting that one, but expert ROV pilots. Successful sample of the seabed. Okay, next up we're going to collect a sea pen that's quite close to SuBastian. This is for Jeremy Horowitz, our black coral and other deep water biology specialist on the survey. So you're currently watching the live feed from ROV SuBastian, which is located 140 metres below research vessel Falkor. Falkor is the Smith Ocean Institute research vessel. We're conducting research cruise in the Coral Sea Marine Park. The cruise is titled the Seamounts, Canyons and Reefs of the Coral Sea. Major activity is ROV dives, margins of a few of the reefs. So today on the northern margin, large reef system, Herald Cays, with a water column and collected samples and jellyfish owned by Dr. Dhugal from Japan. Advised as we move down through the water column by those samples. Now we're on the seabed and we're about to begin our ascent back up the margin of the seabed. Before we begin our ascent, so this survey, especially the thick component of it, is very much interested in acquiring the high-resolution images and samples to help us learn the influence of margin geology and geomorphology on the distribution of biodiversity around these reefs. Are there patterns of zonation, biodiversity around the reefs that matches up with physical characteristics of the seabed in terms of its geology and its geomorphic features? Is there a number of different types of lithology that make up margins and base of the reef? Textures they form, whether it's steep or cliffed or overhang, mantled by unconsolidated sediment. That sediment's sandy or very coarse rubbly. And also we're gaining insights into the processes that drive the evolution of the reefs. Failures, pebbly deposits that line the flank of the reef, and also the patterns we can see seen so far is slope failures. Work probably multiple times down the margin reef, but also some have become very, very heavily cemented and more permanent structures. Also see in the unconsolidated sediment that some currents are quite effective in moving sediment down the flanks of the reefs, and that is also eroding into the ore deposits. But the geomorphology of the margins of the reef is starting to give us some really good insights into some of the major processes that are involved in of the reefs. So now that we're doing a biological collection, I'll pass it over to Jeremy Horowitz, who's part of the science team here, and Jeremy's from JAMSTEC. Thanks, Jeremy.
Thanks, Brendan. All right. Hi, everyone. So yeah, we came across this bamboo coral, which is it's a soft coral. So they have these repeated nodes of gorgonin, gorgonia, soft coral. So they're made of gorgonin and calcium carbonate. And so that's what you could see at the base of this colony. You can see the skeleton has those repeating nodes. So we're just trying to take a sample of this colony. And for the soft corals, we don't need to take the full colony. And what this allows is for the coral to actually continue growing, because each one of these polyps is actually an individual animal. So removing some of these polyps allows the other polyps to continue to survive and grow and reproduce, and it will just continue growing where we just took off the top bit.
Was it you or was it Ben that said he's got I told that one about it, and then
So this bamboo coral, bamboo corals in general, are of great interest to the Smithsonian Institute. So coral curator Andrea Quattrini is working on this group, and similar to what I'm doing, I've learned from her and Pete Cowman, ultra-conserved elements, which is a next-generation sequencing method of understanding the evolutionary history of various coral groups. So she's going to be excited about this this sample. So we're at 640 meters. It's about 6.5 degrees Celsius. And yeah, we got a turtle along for the ride. And although you may think it's a deep descent for this turtle, fun fact, the leatherback turtle can dive down to 1,000 meters. So this is about halfway the depth at which some sea turtles can dive to.
What box was that in?
Q4 before 4.
And yeah, that little crustacean was showing off while we were recording the colony. It'll make for some great footage. Yeah, Reynard. Pete Cowman was working on fishes. It still is working on fishes and ultra-conserved elements, but we've taken him over to the dark side, and he now works on hard corals, and I'm sure he's very happy to admit black corals as well. Yep, we'll head forward. Sorry, Lisa, we'll get you a view of the turtle on the next collection. No worry, he's joining us for the ride. So we saw some nautilus shells on the floor, which isn't surprising. We expect to see nautilus, well, there are lots of nautilus in the area. If you recall, when Rob Beeman was narrating on the last expedition, we saw lots of nautilus, and so hopefully we'll see some during these dives.
You did see a nautilus shell somewhere?
Yeah, just before.
Alright. And I think it,
Oh, you mean earlier also?
yeah.
Yeah. What's the difference between black corals and soft corals? Glad you asked. So, black corals, although they actually look really similar to soft corals, especially the whip black corals and the whip soft corals are nearly identical. One of the differences is that soft corals are octocorals, and they have 8 tentacles, and black corals are hexacorals, and they have 6 tentacles. They're also made of different material, so the black corals are made of chitin, which is a protein, and soft corals are made of gonin, which is shrimp. Actually, the black corals are more closely related to the hard corals than they are to the soft corals. And that's actually something we only realized, not too long ago, when others were understanding the evolutionary history of the Anthozoa order, the corals. I see. Someone who is interested in knowing. Someone's interested in knowing the history of why there's a sea turtle on our ROV. Anyone know?
It was a picture op for our outreach's daughter.
Oh, okay. Yep. So it was a photo opportunity for someone ashore, and I kind of like it.
Yeah, I think you like it.
Yeah. Actually, my wife studies sea turtles, so I would like to say it's because I was thinking of her and, wanted to, have something remind me of her when we're diving. I should have probably started with that.
Got moving there, Eric.
Another roll, thanks.
I'll probably just keep you going south-southeast unless there's a reason for us to stop, and I'll let you know if we do that. Okay?
Alright, south and southeast.
Yeah, we're still pretty close to Cairns. Yeah, we're still offshore of Cairns. Yeah, anytime I throw out a sea turtle factoid, it's because my wife messages it to me. She's the expert, not me. Can we see, just I guess quickly pan over to there? See what that is. I'm just curious if it's, nah, never mind. Oh, yeah, I thought it was something that's swam. Oh, I think I know what this is. All right, this is certainly something we want to collect. So we'll just settle down.
Sure. If you just zoom up just a touch from Jeremy, I'll just see what's happening.
Zoom out.
Yeah, just because they saw it going.
Yeah.
Is it a black coral jar?
Yes.
500 just past it, so.
All right. So what we're about to collect, I'm really excited about this. Years back, when they did the first dredging expedition of the Coral Sea, they collected these black corals. This species was also collected. Oh, another little crab hanging onto it. And so it's in the family Cladopathidae, and it looks like Heteropathy is the genus. So we have some old samples from the Coral Sea. But the DNA was so degraded that we couldn't sequence the DNA. And it represents a new record for the region, which is really exciting. So actually, can we swing around to look at it face on?
Like 90 degrees?
Yeah, we'll look at it. Okay, a little bit. So this is an amazing opportunity to add this to our phylogenetic tree.
Here, down here. We'll just come to a pause when you get to that waypoint.
All right, holding there. Thanks.
So when we swing around to get a better view, we can explain the different morphological features that make this genus different than the other genera in among the black corals.
Is that pretty good there, Jeremy?
A more
Right from the backside?
from the front there.
Yeah.
Well, this will be, yeah, when you settle down, you'll be right in front of it. Yeah.
Shall we just start collecting while you're talking about why it's so special?
The whole thing?
Yeah. Okay, we're just going to settle down and take a look at it, and then we're going to go and grab a sample. Okay. Yeah. We're down?
You're in manual now.
Okay, thanks. So we're just gonna get a nice image of it in the water. It's auto.
Just turn back on and try that.
Beautiful. Okay. So we'll wait for it to shift back to us. But the pinnules should be sub-opposite, so on either side of the stem, these branches, and the repeated branches, so they're pinnules, will be just offset by a little bit in each row. And you could see also pinnules along the stem. So we could zoom in on that. So right where the crab is, you could see he's kind of standing on some short pinnules. That's beautiful. All right, we're just gonna zoom out and do one focus view with the lasers, and then we'll grab it. Okay, so it's not too big. Okay, we'll grab it. Okay, so as I was saying, this specimen is really exciting because it'll be the first time we're able to add this genus to our phylogenetic tree. And what this will allow us to do is understand how it's related to other genera among the black corals. Although we have samples, none of them were viable for DNA analysis, DNA extraction and sequencing. This is about what you'd this depth at which you'd expect this genus to grow. Certainly deeper. Yeah, you can grab the whole thing. Yeah, absolutely. Thanks, Rob Beeman. Yep, so the expedition that first collected this specimen, this species, was on the Sedaris voyages from 1986 to 1992. And they provided us a whole lot. Is that a nautilus shell?
Yeah.
Oh, man. Aren't we getting a treat here? Yeah, let's get a good image.
It's very fragile. Yeah, it's very fragile.
Yeah. All right, so I'll just focus. Can I get the autofocus?
It's off right now. Oh, you want auto?
Yeah.
Yeah, it's on now.
Okay.
Let's not talk about that.
Or you want manual?
Manual, sorry.
Yeah, there you go.
Okay, so just focus on this beautiful image. So nautilus shell, black coral, and a little critter.
I have a ton of frac crabs.
Yeah, okay. All right, we can get it in. That's all right. Yeah, if we can preserve the entire colony like this, and so when it gets back on board on the back in box 3-4, if we can preserve it as is, I'm going to do my best. Yeah, we could try to maybe display it at the Museum of Tropical Queensland because there's a whole bunch of amazing stuff going on right here. And we have to ask the sea turtle permission. Is it allowed? What's the name of the sea turtle?
Terry.
Is it Terry?
Terry the turtle.
Terry the turtle? So Terry's debating whether he allows us to insert our specimens.
I can if you want to. Yeah, I'd rather because it's really brittle, so I don't want to have one. Yeah,
Yeah, I think this is the first specimen representing Cladopathidae that we've collected. We know it's abundant in the Coral Sea. We needed this specimen, and now we've got it. And a Nautilus shell to go with it, because, you know, why not? No, it's not an alive sea turtle, sorry. Although it does move. Yeah. All right. Now that we've collected that amazing specimen, we're going to head towards shallower water. We're going to go up the reef.
Artistic representation.
Beautiful. Not your first part.
Back on track.
Yeah.
Voyage ROV SRG, you're moving it.
Update, thank you.
Get some push cores as well. Yeah. Each study you want to be honest.
Watching the Fungi Beam acquisition this morning on the team here.
The devil's Delta. Don't do it. It's there. Alright, go for the chase.
Yeah, who would have thought the coral was growing on a Nautilus shell? It's amazing. It really is. So we're just heading forward, trying to get towards a slightly different habitat. We want to get shallower, where we'll see different coral assemblages, and we would like to take some rock samples as we move towards the top of Herald Cays.
Oh, yeah, 20 meters.
Shannon says hi to me. Said it, are you getting a chance to look into Alanita beds?
So we're just exploring, looking for interesting things.
Hey Adam, can you sing out when you're getting to waypoint 3 so I can log? That's when we'll start doing the 500 meter transect.
Yeah, we're still not even at waypoint 2 yet. A little bit yet.
Thank you.
But I'll let you know.
Scoot along.
Yeah. Beacon marker.
Yeah, thanks Rob Beeman. So those long feeding trails, I'm not going to pronounce this correctly, Lubenspuren, which is German for life traces. Ridiculous.
Yeah, yeah.
Just,
Yeah. Because he's supposed to come this way. No, you got down.
I think someone was asking what we will do with the images. So on this voyage, on this expedition, in addition to collecting substrate samples, rock samples, we're also collecting biology, and we're taking images of what we collect. And this provides us an opportunity to see exactly what the specimen looked like at depth, also what habitat it was in, what sediment it was attached to. And often it's really hard to get good images in the lab when it's kind of lying on the table. Often the best picture you can get is when it's still in the substrate, sticking out of the ground. So we'll take those images and combine them with lab images that we take, and then with all the other stuff that we do with each specimen, which is, record, document the suite of morphological features, sequence the DNA, and combine all of those things to understand that specific specimen. I know there are people. Lisa, the unknown explorer, often posts lots of screenshots of what we find. And here we see another nautilus shell. So there are probably plenty of nautilus shells in this site. I imagine we'll see many. Some fish. Another nautilus shell to the left. Can we go twice as fast? I'll get to the...
Yeah, just have to
Oh yeah, no problem.
catch up there.
I'll tell Eric we're going to try to speed up a little bit. Bridge ROV.
Bridge.
We're going to try to speed up a little bit down here. Do you think it's possible to ship a little faster, or are you maxed out?
Laterally, pretty much maxed out.
That's right. Okay, thank you.
No worries.
Betty, so you missed a pretty exciting beginning of a dive. Yeah, definitely scroll back a bit and see the amazing things that we saw.
Notifications from Slack.
All right. So it seems the sand habitat has ended, and now we've got some rocks, some spider crabs. Some little corals, maybe a little soft coral there.
I just see a wall here. I don't know what it is. It's breaking to a stop. Just because I don't know how that is.
So the age of the coral we collected earlier, we don't know. We really have little understanding.
Right here, we just hit a wall. We're gonna climb up it. We don't know how steep and how high it is.
We've little understanding about how slow these corals grow. We're definitely aware that they're slow growing, slower growing than shallow water corals, but even between species, the growth rates vary, and it's really hard to determine the age of a coral even when it comes on board. I mean, the size in general doesn't tell us, because there are big corals that can be young and small corals that just have a maximum height of 10 centimeters, but they could be really old. So we don't know. There have been studies about trying to look at the growth rings. So similar to trees on land, black corals and bamboo corals, actually many corals have these growth rings that relate to seasonal changes. So you can determine the age of the coral from looking at those growth rings. However, after looking into it further, it's not always so easy to determine age from growth rings. It's not always so clear. So at this time, we just really don't know much about how fast these deep sea corals grow.
Yeah, that's good. It's just close by. These guys are interested in my book.
Do you guys have any rough idea of the history of sea level change around here?
Okay, let's see another bamboo coral. We'll pass by it for now, because we collected one earlier. Oh, I thought it was zoomed out all the way.
That's what I mean. Do you think this is actually like a wall, or it's just falling near the surface?
Another big whip coral coming up here.
How are we doing with the ship?
We paused him just because we don't want him to run over top of the cliff and drag our boat around. So we're just basically kind of straight up here.
Great. Okay.
I mean, we see that hydrogen being provided as tools or seabed.
That looks like. Not sure what that is. Maybe it's a black coral? No, it's another bamboo coral. Okay. So we're seeing a lot of these bamboo whip corals. I haven't seen any bubblegum corals yet, but we expect to find them.
$20 if you can't find you, okay?
So we're just rising over these rock structures. Looks like a sea pen.
Very spectacular.
Just quickly zoom in on this white coral. Okay. Just gonna zoom in on this. Oh, it looks like a little cup coral.
With this pin form that shallow.
Zoom in.
Start below you. Start below you. You see it? Right here.
So we're going to collect this A. zooxanthellae cup coral. So it's a scleractinian single polyp coral, and there's a lot of interest in this coral among the scleractinian experts. So since we've seen it, we should grab it. It'll probably be attached to some sort of small rock, so you can probably grab it at the base.
Yeah, we'd like to try and get a rock as well. Sure.
So while we're here, we're also going to try to get a rock sample.
Yeah. And get cover for the coral and fork for the rock. Yeah.
Can't you do both at the same time?
I'm gonna try to get a rock on the. I'm trying to get a rock and I get the coral, but then I can pull it right up and turn the piece off.
Yeah.
So big coin or.
Yep. So collecting cup corals, and actually many of these deep sea corals provides insight into previous climatic events. So actually one of the more common corals used to understand, what temperature and salinity and other conditions were like thousands and thousands of years ago are the bamboo corals. They're very slow growing, and they hold all that information in their skeleton as they grow. So when you collect a bamboo coral that's potentially hundreds or thousands of years old, you now have information of what those previous conditions were like. And of course from that you can tell how it has changed until today. Oh, there's many of them, but
Which one do you want?
The bigger one.
Alright. Do they snap off pretty easy?
Yeah, one the other day was very tough.
Yeah, I guess try to grab it at
the
base. These are not squishy, so these are made of calcium carbonate, scleractinians. So they're hard, they can break pretty easily, and you could see at the outside of the polyp there, you could see those structures. Oh, that was perfect. So you can grab that right there at the base.
You want us to hit it with the such and
Sure, if that's easy.
Whatever y'all want.
Whatever's fastest and easiest.
Try to pick it up real fast.
Okay. So the characteristics of the top of that polyp, the skeletal structure, are one of the more formative features in identifying the species. So from our last expedition on the ROV Investigator out in the Coral Sea as well, we collected, I think, maybe 15 cup corals like this. And we sent them to the Smithsonian Institute. An expert, Stephen Cairns, scleractinian expert, was able to identify the species, and he found that some of them were actually undescribed. So now we're able to see what we're collecting, and we can collect living samples, and that will allow us to, you know, better describe the new species. In my box, you can put it in A.
Look at my head. It's not a bad thing.
Can one open?
Beautiful. That was a really good job, guys.
Go. Alright, we'll go to the other arm and try to break up the rock.
Okay. So yeah, we're going to try to get a rock sample. If you remember from previous dives, the rock is really hard, and this looks similarly hard. But science is about trying. So we're picking out a rock that seems to have enough surface area for us to get our claws around it.
The area where we just pick the coral, that's not necessarily part of the that's right. At the very least, the ball is careful on that ledge.
So you can see the RV has these two, I guess you can call them bumpers, which allow the RV to push forward against the rock and then therefore have the ability to take an arm and pull backwards. So it, because the RV is neutrally buoyant, it's hard for it to settle down and pull up. But in this way, it could push forward, not damage what it's pushing against, and grab something backwards. Are these bumpers a new addition to the RV?
The edges,
Yeah.
The rails have been there. We usually use the rails to pull things like core tubes. But you just put the two ends on the rubber.
Yeah.
More.
Yeah, it's a new addition to the RV. What are we calling them? Bumpers?
We call them the left kisser and the right hook.
Rock and sock 'em.
You heard it here first. The left kisser and the right hook.
So we can. Yeah.
Gold color. Make it gold. What color you got? Oh yeah. Let's do some blue. What colors they grow in the dark? Beyond the red line. Cheers. Awesome.
I think people like Rock'em Sock'em.
Rock'em Sock'em.
Oh. What does it mean?
No, I'm just looking at the comments.
Ah. Just a reminder, those two green lasers are 10 centimeters apart. And yeah, the Rock'em Sock'em does it again. We got a rock. And
One, two at the same time.
it's a pun. Oh, that's clever. That's clever. I think we want to look at it, right?
Yeah, can we look at the larger place?
So we're going to look at the part that broke off to kind of see the inside. So
Open up the.
it looks like it's made of
Okay, great, thank you. Let's stick it in the box. Do you mind if it goes in 1A, Jeremy?
yeah, throw it in 1A.
Yeah, 1A.
Okay. So Marty, does it look like it's made of corals?
I'm not sure coral, but looks like it's limestone.
Okay. So Marty reckons it's made of limestone, but of course we have to look at it back in the lab. Similar to everything we see and collect, it's really hard to tell until you have it in your fingers. And even then, it's still hard. Yet, I think there are worm tubes
Yeah, that's what we thought.
inside. Good eye. Yeah, even after the entire trip's over, it's still hard to tell what we've collected until we get it back in the museum and do our various tests. For example, right now in our wet lab, we have 7 large black corals hanging from the ceiling, and I'm still trying to figure out what they are exactly.
Sure, one's open. You know where that cup from?
1A, right?
Yeah, it's okay, thank you.
Yeah, I'm sorry.
Okay.
Beautiful job.
Okay, continuing on.
Yeah, let's do it.
Alright, I'm gonna. You should have a little bit, or you.
Let's just wait till that sonar opens up a bit, just because I don't know what the depth is going to be.
Looks like a flat good back.
It's right on this.
Alright.
Sorry, not the soft one.
Rockwood soccer man.
So, rocket or soccer?
Sure. Yeah. Rockwood.
So Rockham, okay. Yeah, I'd like Rockham to be on the right.
Rockwood, all right?
Yeah, it makes it a little easier for me to remember.
Okay. We can do that.
So Rockwood, you're down.
So Rockham is the right bumper, and Sockham is the left bumper.
A good perspective of the face of the mountain. So if I look down at that camera right there, it's just the sheer.
Yeah. So where can we change that view for the?
You show somewhere.
So we're about to change the view for you, so you can see the sheer drop of this wall. We're going to shift to one of the other cameras. Yeah, it's pretty amazing. So we're just trying to get the right image.
That's a good one there too. You can see down the fourth side of the wall.
Either of them. Yeah. That's all right.
Stay requested.
That's okay. Next time. All right. We're going to have to continue forward. We'll try to get that view later. So I see some more sea pens. So by untrained eye, similar to the one we collected, the one we saw earlier in the dive.
Bridge, ROV.
Alright, Bridge.
I'm gonna move you down south a little bit. Nice and slow though.
Alright.
Thanks, sir.
So we're now at 575 meters, 7.7 degrees Celsius. Herald Cays.
Jeremy, you're asking about things that we'd like to fly the ROV on. This time,
This is it? This is cool? Yeah.
Subsea canyons are cool.
Yeah. And what makes it so... you want to... Can you get a mic? You want to talk about... So the ROV pilots were saying how they love flying over this types of habitats. Why is that?
One of the things about this is that it's interesting to look at the different geography and the little caverns and the rock formations. Not that sand isn't amazing to look at as well, but it's something new. Plus, it's a bit more challenging. You have to be aware of all of your surroundings, both on the side, above, below, make sure you're not flying into anything. Then whenever we land out to try to make a collection, that's also a new adventure to try to figure out the best way to do it, the safest way to do it, to get the collections inside and so on. So it just makes it a more interesting day at work.
Thanks, Chris. Yeah. So what we see here are glass sponges, a whole bunch of them. They seem to be about between 5 and maybe 8 centimeters in height. And so these are made of pure silica. We collected one really amazing glass sponge the other day. I think we'll hold off on how hard is it to collect this. It's kind of odd. It's at a hard angle.
Are they pretty delicate?
Yeah, you know what? Let's do that later. It's a tough position to get at them now. We'll have other opportunities to collect this, so we'll just continue forward. Yeah. Yeah. So we have. Yeah. So we have about 2 more hours on this dive. So we're going to try to make our way up this seemingly sheer wall. So as we're going up, once again we could look down at what's below us, and you can see this rock cliff. I think it's on your screens now.
Just pull that picture to the over and pull that to the screen there, right? There you go. Now they can see the view.
Yeah, so it's amazing to look down at the maybe 50 meters drop, something like that.
Yeah. Or even.
Pretty amazing. Okay.
There's a question how many cameras they have on SuBastian. We have, not counting the...
Counted everything with the GoPros, it's 12
with the GoPros.
Yeah. So we have about 12 cameras on SuBastian right now, and so that consists of the normal cameras on SuBastian and the additional cameras that we put on for our specific scientific studies. So that includes looking at the jellies in midwater. So we have GoPros on SuBastian as well. So we have plenty of footage of every second of this amazing dive, which we later go through in high resolution. We compare what we collected back to the video and also the images. And the next few papers that come out from these expeditions will have these images in the figures, because really you can't beat the images that we're taking right now when we are next to these specimens in situ.
Just a thought, because the way the ship has to set up this location, facing this direction, in case they get blown off, they don't get blown away. So just give us other patients. So fast down. Just looking at the next waypoint, just looking at 2 hours or so. You want to start transiting a little more, because it's going to take us a little bit. We can't outrun the ship. So we're just going to do it at this speed. Do you want to start to transect a little bit before that, kind of maximize your transect? That's probably a good idea. So we're going to talk Yeah, just let me know. We'll keep playing obviously this stretch.
So we've now hit kind of a plateau of sand on top of that rock wall face. So we're just continuing to explore, heading towards shallower reefs.
That's that wall.
Okay, so it's also here, so it's that on top of it.
We do have, you can see in this see the start touches it here. There's going to be some rubbly stuff going on, and I suspect it's going to keep coming up, so I may not be sand the whole way, we're just in a little bit of a plateau, but I'm just not going to let that wall is short.
Yeah, I can look you down south again. Maybe just
Thanks.
Yeah.
Okay. So yeah, give me at least blog posts so it's
Yeah, do you want me to pause here?
No, man.
Just keep going.
Yeah. Good.
Sure.
Daniel, let me get back to you. I think we can throw up a bathymetric image of the site. Let me ask for that, and yeah, we'll try to get it up shortly.
We'll drop the waypoint as soon as they start.
Oh, yeah, please.
That begin.
Yeah, there's no sea cucumbers. I guess it's surprising. I'm not sure. Yeah.
Surrounded by, the distance and safety data.
Cody
Yeah,
J.
yeah, we're going to get Falkor on it now.
So, what speed have you got?
.3, .4-ish. 0.3.
Yeah.
It's limited on him though.
Yeah. I'm good when you guys are.
You happy now?
Yeah, thanks.
Started now?
Thanks, guys.
So at this stage, we're going to start our 500 meter transect. And if you'll remember, we did a transect at Cairns Seamount as well. And the purpose of these transects on these different reefs is to have some sort of quantifiable area, so the exact same distance in different habitats, while hovering at the same distance off of the ground with the same camera focus. You can then identify what species and how much of each species are within those 500 meter transects and compare them at different sites. And this will allow us to compare that information with the abiotic information, the sunlight, the temperature, the current, the salinity, and all of that information can tell us what allows an area to have either a high diversity of species or a low diversity of species, or a few, so low diversity but a lot of one species. So answering those fundamental questions, what drives biodiversity, is a really important question, because then we can decide how to protect biodiversity. If we understand what allows biodiversity to occur, we can then, focus on those areas to protect. But as of now, we don't really know which specific suite of features are suitable for high diversity. Generally, we know, but there are transitions from the deep to the shallow and along a latitudinal gradient. So these transects will provide insight to us better conserving biodiversity in the coral sea.
Yeah, so Andy, basically waypoint 4 will be the 500 meter for the end.
Yeah, I'm hoping it'll probably be over more of this sort of stuff as opposed to over it'll be over more of this sort of reef and sand, this sort of thing, I guess, as opposed to sort of reef.
Yeah.
You want to get reef and sort of further on, which is what I was trying to capture, but at least we get it in. So.
And we can get the 500, and then once we have that as a base, we can see if we can get more more interesting heads later on.
Yeah.
I was to go in the 500 and then just cut the first 100 off.
Yeah. We get the 500, once we have that, then we can splice the highlight reels.
Well, that was surprising.
I mean, it doesn't. It's not just sand. Right? We still characterize this as sort of form.
Oh yeah. Well, I'm seeing little corals.
All these tracks in the sand is.
Well, they're still sea hens.
Still quite on.
Pretty durable.
Yeah.
So we just start the 500 meter transect. So it's going to try to keep it nice and steady, going down the same direction. So we get to the end of that, we'll figure it out from there. Deal.
Awesome. Thanks.
All right.
Okay.
Okay, yeah, but I just had a lot of problems too. So I get really when I said to the player, he's like,
What do you reckon the transit from there be?
Pretty large.
Oceans or cucumbers or?
Some pretty large gastrophytes. I mean,
So where are they now? Are they go in?
or versions that are
Super accurate.
there's a couple of versions with some of those really long spots.
You see the frequency change?
So we're seeing here some whip corals. They look pretty red. Maybe they're gorgonians? Oh, no. Look like whip black corals. No. We're not going to zoom in because we want to keep the transects, the 500 meter transect as, undisturbed as possible. But these whip black corals are really interesting. It seems that although they're very simple, they have very few features. So, there's no branches. They have either one row of polyps or rows of polyps around the stem. And they have these really small microscopic skeletal spines. But although at the surface they all look very similar, after sequencing their DNA, this was back in, I think, 2013, we realized that the deep sea black coral whips are very different, so they're very distantly related to the shallow water whip black corals. And what that tells us is that this morphology of no branching has separately evolved multiple times over evolutionary history. That's cool because you would assume that, the oldest coral in a coral group might have simple features, not very complex, and then they've evolved to become more complex. But that's not always the case. It's not always so simple. I guess what drives evolution in that way, maybe to be unbranched, is because the habitat at which it grows is just suitable for a simple featured animal. So although they look the same, they're actually very different. So the morphology can be very misleading. It's called convergent evolution. And so what that tells us is you can't just look at the features of a coral or an animal for that matter and understand how it's related to other animals. You need that second line of evidence, that molecular relationship between the two organisms and all the organisms in these different various groups to categorize them, as we as humans love to do, and understand how they're related. So these whips actually maybe look like soft corals. They're just too orange to be a black coral. And it seems like even if between the polyps it's also orange, bright orange. Yeah, there's lots of really good examples of convergent evolution. There are lots of winged animals that, they must all have wings, they're closely related. But no, it's just that, separately over time, different animals have evolved the ability to move in the air. And, they may look very similar because they need to be aerodynamic, have, wide wings. But, it's not the case. So morphology can be misleading. We're seeing here soft corals and what looks like a hard coral in the middle. We've sampled a few hard corals down at these depths, these branching hard corals. They're very interesting. So we'll send those off to the experts for identification. Yeah, another good example is the stalked crinoid, which it kind of looks like an umbrella. We've seen a few of them during these previous 2 dives. There's also a soft coral that also has branches that come off of the top like an umbrella. There's also a black coral, a genus called Umbellapathes, which, as the name suggests, looks like an umbrella. So once again, these are 3 very, very different groups that on the surface look fairly similar. Yeah, it's very interesting. So now we're starting to see a lot of soft corals, small soft corals. There's a coral to the right.
So
Thanks.
Cairns on the west and the Reefs in the east.
Okay. So we were able to overlay a bathymetric map of where we are. So we're off to the east of that image. So you kind of have some sort of idea about where we are and how the depth varies around the site. So we just leave it up for a minute. So why are black corals called black corals? Good question. So they're made of a protein called chitin. It's the same protein that beetles are made of, and so these beetles are black, jet black, similar to black coral skeleton. So the skeleton is black, but then the animal that lives on it, polyps, are different colors. So when it's alive, it won't look black. It'll look like whatever color the tissue is. So it could be white or pink or red or yellow. But when it dies, and the tissue falls off, it goes away, what remains are these beautiful, shiny, black skeletal structures. And that's why it's called a black coral. So the order is Antipathidae, which translates to against disease. Many years ago, a long time ago, different cultures, actually many different cultures, who didn't interact with each other, came to the conclusion that these black corals can ward off evil spirits. So antipathies, against disease. So I know in the Philippines, they would hang black corals over their doorways. So in many different cultures, they're used as a symbol of protection. And it was thought to maybe be some sort of a medicine.
That was what?
I saw it. I saw the guy. Yeah, he's swam underneath us.
So a ray just swam underneath the ROV.
Must have been good.
We'll go back at 2 o'clock.
Okay.
You can come back here at 2 o'clock. 1:37 and 12 seconds.
As the turn, you're going to go over the edges, or on that edge.
Yep. Here.
So there's just a little bit of a drop off to the left of us. So I'll just kind of skirt the edge and get images of these.
Okay. Sure.
Blue water.
We can do blue water, but might not get much of that.
I imagine that's not what's. I'm sure it's not ideal for the survey. Right.
Spectacular, that.
Yeah, it almost feels like you're scuba diving, in the site.
Little sponges. Are they? No, corals.
Yeah, and soft corals. Looks like a bamboo coral, a whipped bamboo coral in front of us.
Which way?
Looks like a crab maybe passing over.
Going southeast, going this way. That Cardian, this headed. Yeah, so yeah. Appreciate you. It's different in Florida than it is in Canada, so that's so
See some fish.
yeah, we know.
Not too far out, because if he goes out there, I'll get too far away from the water. But I think, see this guy here? This is going to be a drop off. So if you put him somewhere like in here, you'll be able to skirt this edge like this.
Come right here. Perfect. Where's your RV?
Another update?
Yeah, more of a Jurassic one this time because we kind of did the
Awesome, thanks.
These those shorts.
I hear you cut off my long-winded explanations.
It's because you put it right in line with the bags. Like, yeah, it's exactly where it went. Yeah,
It's further to the west.
that's you.
So we're seeing small, thin, soft corals. There's a sea pen below us.
Here.
Same as the one you collected? Close.
I couldn't say. I need to get a close look or something, you know, for some other time.
Like, are you gonna come up?
Yeah. I'll be happy if we always get these transects in, you know.
Ooh, another drop off.
Oh yeah.
I don't think it's, I think it's a big problem.
Yeah, we can get more right there.
If you look at the sonar, it kind of gives you. There's life on the other side of it.
Yeah.
Right. Okay. So I guess we just follow it along. Yeah.
Yeah, you can see my contour up there. You see where the darker squares are?
Yeah.
That was kind of like an edge of sorts. So I just had to. Otherwise, you would just been blue water transect.
And so the ridge that we saw yesterday when we were planning this dive, so that's the rocks, right?
Yeah.
So now we know.
Yeah, and so we came up and then we're sort of following it along.
And is that
And the cays here.
and is that what you expected? The rock structure?
Yeah,
That's-
to get a ledge like that.
Yeah.
It certainly looked quite different to what we saw, at Cairns Seamount or Flinders Reef.
Maybe that's why he doesn't do it. We just don't support it. It's like, yeah, see.
Just let him sit down and go.
Yeah.
This question's for me. Do I have a favorite coral? I have a favorite coral group. I probably have a favorite coral species. You might be able to guess what it is. Certainly soft corals. So you got interesting rock structures here. Not really much life. Maybe in those underhangs you have certain critters living where the sediment doesn't constantly flow on top of. few soft corals, and you can see that they're growing at at angles, probably maximizing its survival away from where sediment sweeps across these rock faces. And they're small. It might mean that there are currents that don't allow it to grow to a size of which the surface area would be so large that it would rip the coral off the substrate.
What's going on?
Brutal, I want to set the record straight. Soft corals was a joke. I'm black corals all the way.
It's divergent.
The most interesting thing that I've learned from studying coral I guess just generally how little we know about relationships between species and how new technologies are allowing us really for the first time to understand where they are, why they are where they are, and how they're all related. So for example, the specimens on this trip are going to take us about 6 months to completely figure out everything we need to know about these species and their relationships with other species with a high degree of confidence, where, over the last, maybe 500 years, people have been studying these same groups and trying their best to understand, the relationships between species. So I guess the amazing thing is we now have the tools to completely change, update, build upon those that started these fields to understand the taxonomy and the evolutionary history of these various invertebrate groups. So I think it's more of just in awe of the ability to take leaps in a field. And I guess just feel lucky that we're working on these groups now in this exact moment when these techniques are really just becoming readily available to anyone.
I think there is in speech in place of just
Okay, so we're seeing soft coral. So we're looking over a ledge now that looks like it drops back down
understand the setting.
Yeah. And then it will swing back up, so we're just gonna fly right across it.
Okay. Once I get down, I'll turn back to the left. Okay, that's how that.
Can you just take that bird over that? Send that off.
Thanks, Rob. Identifying the soft coral. The Gorgon head. Rob is kind of knows a little bit about everything. It's amazing to have him on these expeditions. We're excited to have him on board for the second leg of this trip. Oh wow, look at this crab. It's a beautiful crab coming up. Oh, this one, past it. But, yeah.
It's just sand all the way out.
Use the gravity of the rocks to the left.
Which sort of gives full coverage, but it's still a ribbons.
Yes. Yeah, I guess.
Oh, thanks, Rob. Yep, so I'll just repeat that so people on Facebook can also hear. So the ripples on the seafloor indicate near seabed currents. So the way the water moves is reflected in the patterns in the sand.
You can tell Rob that yes, the lasers are on, but the angle of the camera doesn't make
The lasers are on. Oh, you could see it now, I think in the middle of the screen. It's just that it's facing really far ahead of us, so they seem really close together and they're hard to see, but they're there. And we try to have them on for, when we collect specimens or when we're looking at different critters.
Yeah, interesting. With no breaking reef, you know whether we're going to get anything. We're going to get anything common. This is all there is.
Hi, Teresa. That's a really good question. So when we collect our samples, they're deposited in museums, so they are in museum collections. And what can be done from there is anyone can access these museum collections online and look at them. So all of the specimens collected from this expedition are going to one of two museums, either the Queensland Museum or the Museum of Tropical Queensland. But if you search the Queensland Museum, you can click invertebrate collections, and then that will allow you to search through their entire digital collection database, and you can then see every single sample that we've deposited there. And additionally, all of these specimens in some way contribute to scientific literature, whether it's now or whether it's in 5 years from now. I can only speak really for myself, but the black corals that I'm collecting on this trip are going to be used as a part of my PhD. And those images will be in a paper that I will write maybe over the next year, and then it'll get published, maybe a year later. So, that's kind of how the scientific process goes of publishing literature. It tends to take years, from collection to understanding to writing to publishing to getting it out there. So it's a slow process, but it's a good process. It's a peer-reviewed process. So you have to assume, what you're reading is correct at that stage. So that's good. So in the short term, you could see the images in the museum collections, and then afterwards you would see it in various literature. But yet it is hard to, for just anyone to see something be collected and then to know where to look, that's often pretty challenging. But you could always ask. There is a chain where if you ask Schmidt Ocean Institute, I'm sure they would direct you towards the people who are working on the biology during this cruise or any cruise, and you could definitely narrow down where to look to find images of these specimens.
I live in Florida. I think I'd get arrested if I did try to quarantine.
The COVID procedures, well, Schmidt Ocean Institute did take many precautions when with logistics, getting people on the boat. There's also people who have been on the boat for months before we came on, so it requires lots of COVID tests, social distancing before getting on the boat, and social distancing even on the boat. Of course, that's a little bit hard to do when you have this many people in a 90-something meter long boat. It's a pretty small boat. So, you can't always maintain, 1.5 meter distance with people while working on the boat. But we have taken multiple tests before getting on the boat, and we do have procedures in place if something goes wrong. So basically every precaution that could be taken is being taken. And it is really nice being on the boat to be able to do research during this time. I know a lot of people who have had a lot of issues conducting their research. People who are studying in Australia but doing field work in other countries, more or less had to change their entire research project. So for us to be able to go out here and do our research is an amazing opportunity. And it's a pleasure. The weather is so nice. The water is super flat, and the boat is so nice. So, it's really nice working on this ship. How far into the transit are we?
I think waypoint 4 was about 5 minutes. Again, and now as it profiles, sorry, 5 minutes. Oh yeah, we're just like halfway. Under halfway. Mind you, we've been skirting the top of that ridge, so that increases the transect area, but I'm not sure how to quantify that.
They'll figure it out. So we're about halfway through our transect now. You see a little, maybe a sea lily?
Yeah, it drops off to the port side.
Yeah, but not drop off side, it's on the starboard.
Yeah.
We are not allowed off the ship. We're way out in the Coral Sea, so actually really all the expeditions that I've been on where you have large ships doing research that aren't scuba related, it's generally just a no-go on getting in the water. Just too many things can go wrong, and so it's just safe to Oh, there's a Nautilus in the screen right there, but we're doing our transect, so we'll just continue across.
3, 2, 1, go. Basically, we're doing just a transect. You see all the dark patches, we're just kind of skirting into a ridge, so we're not flying in water. And they're going to want to take a rock sample. Whatever she has at yet.
Okay, we got Cody and Ben flying the ROV now. Good question, Brutal. So most of the things that get collected and deposited in museums, they rarely get to see the front of the museum. Often they're in the back end, where they're safe and preserved. So only a select few specimens actually are in the displays when you enter a museum. So whenever you go into a museum, you see X number of specimens. Probably multiply that by 50 or 100, and that's how many specimens the museum probably holds. But certain specimens, of course, will be displayed, whether and it's probably because they represent some sort of new information or just are a spectacular specimen. What we collected this morning, the black coral and the Nautilus, I'm certainly going to suggest that it gets displayed. It's really awesome. And also there's usually shifts in what is displayed depending on whatever themes are being pushed in the museums. So in the Museum of Tropical Queensland, there's a mesophotic section of the museum. So a lot of the mesophotic specimens that are collected are put out in the front to display, what are all the corals that are found in the mesophotic out in the Coral Sea.
Yeah, we should send all of it. Yeah. So it's like a cross.
We're in the northwest of the Cay for this dive. Yeah, so we have the display there. Yeah, so we're doing a transect we started at about 600 meters, and we were working our way up the northwest side of the Cay.
Yeah, I'm enjoying it.
What do we do with the live specimens, like the jellyfish, etc.? So we preserve the bodies. So first what we're doing, and this was based on advice from Dhugal, who's working on the jellies.
That's right.
We subsample a tentacle in 100% ethanol, and that'll be used for molecular studies. And the rest of the body gets preserved in formalin. And formalin is a fixative that preserves all the tissues, generally, as it was when it was brought up to the surface. And that's different than, for example, ethanol, which also preserves the colony, but it can modify the tissue structure. So it'll suck all the water out of the animal. And so, for example, for corals, it will close the polyps up. So after it's been preserved, it's hard to see what those structures look like. So the lasers are always green, and they're always 10 centimeters apart.
That's what I was doing on the SuBastian logo. I'm gonna do another white balance. It looks very, very cute.
Yep, it does. Yeah. So we're just gonna look at our SuBastian logo to get some white to do a white balance. We for the jellies that we collect, we don't observe their behavior on the boat. I wouldn't even know how to go about doing that. They're so hard to even see. I can't imagine someone recording its behavior. So no, we don't. We just preserve the tissue, or the body in this case, and put it in a freezer, and then we'll give it off to Dhugal to work on after and do all the amazing things he's doing, like describe new species and expand the ranges of known species, things like that.
Going on transit?
The jellies, they do get looked at under a microscope. So we have a USB microscope on board, which is pretty good. It's really hard to bring microscopes out into the field, but I have this nifty microscope that plugs into my computer, and you can take a really nice macro shot. So I'll use it for small samples to look at polyps.
Is that 12:30?
So we're still doing our transects. We're getting there. We're in the second half of the transect, so it's almost over. And we're still working our way up to the shallows, although it's taken a while for us to get up into the shallows, so we're probably not going to get all the way up on this dive. That's right. Yep, the USB microscopes are great to have in a house. I once saw a tick using a USB microscope. It was very useful. A question about a Category 3 affecting the marine environment. I guess I'm not really sure. Obviously, the deeper you go, the less impact the storm would have on the benthos. The deep sea tends to, fare better Just generally, the deep sea does tend to be more stable of an environment, so it's protected from storms. It's also protected from temperature fluctuations. It's such a stable environment that you often get, so we call the deep sea the Museum of Biodiversity, because really old things that have otherwise gone extinct in the shallows, for reasons directly related to, climatic changes that are in the surface, or sea level dropping 100 and something meters. In the deep, the conditions are stable that allow these species to just survive unchanged. So often when you go down to the deep sea and you sample the biodiversity, you can be getting much older specimens that aren't found elsewhere on Earth.
Sorry? Joydica, I'll check. She's our deputy director.
Hello, Joydica.
57 meters left, Jeremy.
Hello from Falkor. Sorry?
57 meters left.
Okay. We are nearing the end of our transect.
So what do you want to find, Jeremy? What's on the bucket list?
Ooh, what do I want to find? Well, I guess in this specific dive, our main goal is to try to see what's in the shallows.
Okay.
But generally, we want to sample some black corals, some soft corals, some sponges, and hard corals.
So
No, those are just the target groups. Yeah, no, there's a lot that we're not sampling. We're not sampling fish, just because no one on the boat is particularly interested in them.
Definitely targeting rocks from around 130 meters.
Yeah, if we can get up there.
we'll get up there.
Or Marty, we'll get up to you.
I've got faith.
If we get there.
Oh, we're gonna get up there.
We're going to get up there.
So is the slope starting to get a bit more drastic soon? Yeah,
Feels like it. Yeah.
it feels like it. That's all I need to hear.
Since I sat down, I came up 30 meters.
Right. Yeah, and we spent hours at
Yeah.
600. Yeah, so it seems like we're starting to climb a bit more now. And Rob Beeman suggests it's possible that the boulders found on steeper flanks of these reefs were mobilized by cyclones. Looks like another bamboo coral passing. So now we are at 486 meters and 9.2 degrees Celsius. So getting a little warmer. Certainly not warm enough for my liking. There's a fish.
I'm just following.
Should be over there, Jeremy.
We're nearing the end of the transect?
Yeah.
All right.
Once we hit this big boulder, we'll call it.
Okay.
Gives us a little extra thing just in case.
Yeah, sure. We can stay down for a dive. We can stay down as long as we want, really. What limits us is the lack of sunlight. So it's a bit dangerous to drive the ROV at night when you can't really see. Deploying it and retrieving the ROV is a bit of a delicate procedure. I think we'll just keep going shallow.
Okay.
Nonetheless.
Pretty specky boulders there.
Yeah. So, technically we can do a 12-hour dive, and we actually might do some really long dives on the second leg of this expedition. So currently we're doing, we're also doing AUV dives. So.
In the transit.
In the transit, yeah. So at the moment we're sharing the time with other operations that are going on, but during the second leg of the dive we'll be more focused on the ROV dives, which will allow us to be in the water much longer and spend more time to look at all the different critters we're seeing and go off on tangents and collect down in the deep, and then have the ability to also get to the shallows.
Just a very good position.
Okay, thank you.
Is this a good speed, Jeremy, or would you like to go faster?
You can go faster. So we haven't yet really seen a distinct change in habitat, but similar to yesterday, when we got towards the shallows, around 300, 200 meters, we saw a distinct change where, you started to see coralline encrusting algae, different types of hard corals, sun-loving corals, different assemblages, and there's a shift in these assemblages, and we know there are specimens that occur in the deep, we know there are species that occur in the shallows, but where's that threshold, and what creates that threshold that allows these changes to occur? That's one of the questions that we're going to be answering by sampling from the deep to the shallow. So we're coming up here to what looks like a soft coral. I'm not going to try to identify it better than that. But we'll keep going. Yeah. So at this stage, it's really hard to say for sure when we will be diving. We seem to be diving almost every day, but for the next few days at least, it's really hard to tell whether we are going to wake up and throw the ROV in the water, or if we're going to let the AUV do its thing and then do a dive in the afternoon. So yeah, it's just uncertain at this moment. So we're coming up to, we'll just hold off for a second. Just look at this. It's probably a soft coral. Yeah, looks like a soft coral. So you can see how the polyps are extended really far out. I'll just...
Hey Aaron, just updated your position.
Can I get the manual focus?
Yeah. Then can you click that autofocus button? Take it off auto.
Yeah, thanks. So we see a bamboo coral, and you could see those interchanging nodes of gargantuan and calcium carbonate. I'll just zoom out a bit so we get a better view. There we go. So you can see those nodes in the skeleton. And I'm going to try to zoom in. We're kind of hovering, so it's hard to get a focus view. But you can see the polyps are extended, and you can see 8 tentacles. There you go. That's a nice view. So you can see the 8 tentacles there. And that's right. And the polyps look like they're alternating on either sides of each branch. Oops. So beautiful bamboo coral. I think we should get a sample. Is it all right to settle down?
Sure.
All right.
Why is the stem of that bamboo greener than some of the other bamboo corals?
Yeah, I'm not sure. There's a lot I'm not sure about, especially with bamboo corals. But we've collected one whipped bamboo coral, so an unbranched bamboo coral, and this is of course a branched bamboo coral. So I'm gonna go out on a limb and say they're different species. And so we can grab one of those branches. Yeah.
One of the green branches or the clear?
The green, yeah, one of the green. So it is green. It's just it's more clear near the base.
Mm-hmm.
So I don't know. When you get your arm in there, we could just kind of. I guess I could focus. I'll let the arm come in first, and then one of these branches in the front here. Yeah, so many branches to choose from. So once again, we're collecting a bamboo coral, which is of interest to Andrea Quattrini at the Smithsonian Institute.
All right, if you want to take control and you can follow him.
Yep.
Just zoom in and
Yep.
show him where you want.
Yep.
All right, he's got the sign, so I'll show you.
All right. I guess just come down to one of these, to this branch here. Or, I mean, it doesn't really matter which branch. Kind of anything you can get a hold of is fine. It doesn't need to be so much of the colony.
Can you zoom in a bit? Jeremy?
Yep.
Most birch colony, they want to know what's the progress of the AUV and if we're going to dive with AUV today or not.
There we go. That's perfect. Up. Yeah. You can even come towards the end of the branch a little bit more and get less of it. That's also fine if it's easier.
Hey Eric, I'll have an update for you here shortly.
Yeah, there you go. Perfect.
Good?
Yeah. So, the age of this colony, I'm not sure. So bamboo corals can get extremely old.
And I'll follow that, Jeremy.
So, I mean, I'm not going to try to guess the age of the coral. I'll just be wrong.
What you there, Ben?
We could throw it in bio box 3 and 4.
3 and 4?
Yeah.
Okay.
Yeah. We don't need a close up. It's okay.
Okay.
Thanks. Though.
Yeah, no
The polyps look, I think, white. I'll be able to tell when I get it back on deck. Yeah, so bamboo corals can be extremely old. They also tell us a lot about past climatic conditions. So there's a lot of reasons to collect these bamboo corals. And of course, the main reason why we're collecting it here and now is to understand the taxonomy of this group. And for this coral specifically, we only collected one section of branch, which means the rest of the coral will survive and continue to grow. That won't always be the case, but in this case it is.
Good job, Ben.
Beautiful, beautiful.
Just curious.
All right, you ready to keep going, Joe?
Yep.
Let's do it.
So it's a good question, Rob. Yeah. So the rings of the bamboo corals can be used to approximate the age, similar to the black corals as well. But they also date the corals by looking at the chemical composition of the skeletons.
Amberjack. Look like an amberjack.
I think in order to date it, you would have to get a section towards the bottom of the coral. But again, this I'm not 100% sure. But newer skeleton growth probably doesn't have the information that the base of the skeleton has.
start, I'll hightail it up and try to get to the top.
Up to about,
To the launch day usually, right?
let's go up to about 100.
Yeah, so
Can scoot up to 200 metres.
Yeah, we're just gonna go home.
So Cody, can we scoot your quick up to about 200 metres?
Yes, ma'am. I'll jump up right now. What I'll do is, if you're fine with me, I can go faster in the water column. So I can leave bottom. I can go about right there depth and just beam straight ahead. Try to get.
When we get to about 200, we'll want to be looking at the...
Yes.
Yes.
Okay. Alright, then I'm gonna be going a little faster, so might need to take it out. 200 depth.
So,
Hey Eric, we just updated your position. Also for your information, we're going to go a little faster. We're going to try and make it to shallow water, so if you can speed up your the vessel too, that'd be great.
yeah, let's get up there. So, we don't have much time left on this dive.
Roger that. Thank you.
So, we're gonna try to get up to at least 200. We want to take some more rock samples, and if time allows, and if the right coral presents itself, we'll collect that as well.
Yeah, I think those are amber jacks. Or bull jacks. See how fast they move. What they feel like when you catch them doing. We got around 100 meters to go roughly. That's profile.
Not bad, we can do it.
I think so. Ian? To around 200 meters depth.
So this has been a pretty successful dive. We have done our repeatable transect. We collected some bamboo corals, an amazing black coral on a nautilus shell, which is certainly the highlight of the dive. And if we could just, if time allows, we'll get up to the shallows and at least view, what the habitat is like and what's living in the habitat at around 200 meters. And then, after we sign off, we'll receive the specimens on the deck, and then we'll get to work processing the samples.
Anything adventurous in that thing?
Nah. I mean, yes, but
yeah. Nothing that's really looking right now.
yeah. So the maximum depth of ROV SuBastian is 4,500 meters.
What is mapped? That is the shallowest.
yeah.
Yeah. Shallower or market too?
If you missed the beginning half of this dive, I suggest looking at the video later and checking out what we collected. We collected some really amazing specimens.
Well, there is based on the multi.
Yeah, it looks like red spots were targeted.
But yeah, it's not.
It's not exposed.
That's for the couple of 100 meters. Accidents, and I feel having forced to go faster.
Bridge, ROV.
ROV bridge?
Yeah, Eric, is there a better heading for you to go a little faster?
Alright, I'll try to go even faster than this.
Thank you. Sorry, mate. Updated your position, Eric. Yeah.
I'm supposed to change my outfit to fish islands to get that condensation built up and starts eating away. How far ahead of
Seriously?
That's not
I'll just look around a bit.
might have to come back, actually.
Yeah, it's all right.
Yeah, you know, the shit.
So tomorrow we can't say what time the dive will happen, and it may not even happen at all tomorrow because we also have AUV work to do. So I guess the only thing I could say is I guess just put on your notifications, and you'll know about it minutes or when it actually starts, unfortunately. But as the expedition continues over the next few weeks, there will be more consistency in when the dive starts and ends. It's waiting on.
Just a little bit. Yeah.
Totally. All right, we'll look around. We have to wait a moment for the ship to catch up. So in the meantime, we'll see what's in front of us. Marty thinks it's asparagus. We're just going to hold up here for a minute.
And 3, 2, 1, be ready.
And we're off. Okay, we're flying. What's this button on top do?
Fire some missiles.
Yeah. Then why are we trying to break rock with claws?
That's what I'm trying to say. I think it's supposed to be for to turn on autofocus.
But that doesn't work.
No.
Okay.
It's not matter.
Just so I can pretend it's a missile.
Pretend, that's what I do up here. But it works now, so I can't do it.
You should really get Halo on here and use that as a controller.
I'm telling you, that would be nice.
So are ROV pilots amazing gamers? I imagine you have to be pretty good with, you know, a remote control and...
Consequences are a little bit different with this.
I mean, but in a game, you die. Here, it's just a robot. I mean...
One's a $50 game, one's a $6 million ROV. What's that?
Rock. Lots of good, awesome.
So we just want to do a test, like on the brain, and see, make sure the web, that's what it is. And that will get
So currently we have Cody and J-Rod and John up front here, along with myself, Jeremy. If you want to see what we look like, this is us. Hello. Give a wave. So this is what it's like in the control room. Lots of screens and joysticks. Did you guys play with lots of remote control cars while growing up? Yeah. So still do. So if you're no.
You haven't? That little Jeep
all right. So yeah, if you're playing with little toys, you could be an ROV pilot too. That fella in the back is John, lead tech, who's keeping our feeds alive right now.
Rich. Hey, are we going to stop here or keep on going?
Keep on going, please.
Open. Yeah, so our This keeps going. We're trying to shoot up to 200 meters. Right now I'm at 310.
Yep. So we're in a control room on
start heading up
ROV
around
Falkor, and the robot underwater is a remotely operated vehicle, or an ROV, and so it's attached by a tether to the ship. And
Yeah, I think they're going to try to
at all times, there's 2 people controlling the ROV, and additional people helping with focusing the cameras.
Yeah, we're going to be recording SuBastian.
So yeah, we're not actually underwater, although it'd be pretty cool if we were.
Cool. All right, brother. Good.
There are fish.
Eric, go ahead. And
Three.
so, roughly 50 to 30 ROV conversion and GOV after that. Okay, thank you. No worries, I'll keep you posted. Great, thanks.
So we're at 285 meters. It's now 16.7 degrees Celsius. In a little mini canyon here, chasing a fish. Sandy Lee asked, is there shallow water species that are not vulnerable to bleaching? There are species that have different heat tolerance levels. So, species with wider heat tolerance ranges will fare better than species that require a specific temperature. And then if the waters increase, or if there's a duration of time when the sunlight's really strong and it's heating up the water, so those species might be the first to go by the wayside. There's also tolerance to different acidity levels. And again, groups of corals like hard corals require a specific pH level to survive, which is why hard corals in the past have gone extinct, in past climactic events. But other corals like hard corals and soft corals, which aren't made of calcium carbonate, and therefore their pH level isn't as set in stone, so to speak, they would probably do better in the long run. Coral bleaching is when the temperature increases or the pH level changes. So each animal has a certain amount of flexibility when it comes to the ocean conditions. And for example, when it gets too hot, its symbiotic algae, zooxanthellae, which acts essentially as sunscreen to protect it from the sunlight. If there's too much sun, that zooxanthellae will escape from the coral, leaving it exposed. And then that sunlight will kill that individual colony if that happens. So that's coral bleaching in a nutshell.
passing 250.
Yeah, it's a good question. Well, I mean, an individual coral can't necessarily move deeper, but I think what you mean is, can they, can larvae settle and grow in deeper habitats? So there's also constraints to how deep different species of corals can grow, but that's not the case for all corals. There are certain species that can occur over very wide bathymetric ranges. We're talking about 1,000 meters. So yeah, they do have the ability to grow deeper, or in other words, among that species, the shallow water species will die, but the ones in the deeper habitats would survive, which would make it seem like they moved downwards. So corals that are capable of moving down, if that is more beneficial to that group of animals, then they would do so. The same is true for competition. If there's lots of competition in the shallow habitat where there's lots of sunlight, corals that don't necessarily need as much sunlight to grow have the ability of minimizing their competition and moving away from the shallow, hard coral dominated habitats. So there is this flexibility of different species to, maximize their chances at surviving by minimizing competition or, staying away from the high temperatures. But if you're an animal if you can only live at a certain depth in a certain type of environment, and then that environment changes, there's nothing the animal can really do about that.
$25 meters left, Jeremy. We're almost at 200.
You said you could do it.
We're almost there.
All right. So we're at 220 meters. It's now 20 degrees Celsius. And I think we want a rock sample, right?
Not at 200.
At one.
Start looking, and ideally around 130 meters.
130.
Yeah.
Okay.
A ledge breaking off a bit.
Okay. Did I say 200? I meant 130.
130. Okay. On it. Well, if you see any coral.
Yeah.
Plus whatever Jeremy wants.
Yeah, we're starting to see.
I'll stay at this view, Jeremy, if that's okay.
Yeah, that's great. So we're starting to see corals. Looks mostly soft coral. I think there's a black coral to the left, potentially a Parantopathes, but not a priority for this dive.
No. It's true.
So corals can adapt to coral bleaching. Over time, they can become more resilient, especially if they've survived a bleaching event. And corals also have the ability to change their zooxanthellae assemblage. So that suntan lotion that they use, there are different types, and some are better at protecting the coral from the sunlight. So, for example, during a bleaching event, the zooxanthellae would get expelled from the colony, and then if the coral survives, a new type of zooxanthellae can enter the host colony, thereby making it more resilient in future events. Spawning is not my area of expertise, so I'm not going to try to explain spawning. So we see Gorgonian sea fans, Gorgonian whip corals, little branching corals. If we had more time, I think we would stop and look at all these little branching colonies. There's probably a lot of interesting colonies here. I mean, honestly, whenever you see these types of environments at these depths in sites that really haven't been explored, there's bound to be undescribed species. So in future dives, we will have the chance to kind of sit down and look around and pick and choose what we'd like to collect. So we're seeing whip black corals here. I don't think zooxanthellae would work on humans. So scientists are working on growing corals in labs that are more resilient to rising temperatures. That certainly isn't the final answer to climate change, for example. If we were to grow resilient corals that can withstand temperatures up to 40 degrees Celsius, well, it may keep corals around a little bit longer, but what happens when the temperature increases to 41? So growing corals in a lab seems more like a band-aid solution rather than mitigating the primary drivers of climate change. So we're looking here at what looks like Myropathes, a black coral that is branching in flabellate, meaning it grows in one plane. We're not going to collect it now.
Jeremy, if you see something that you really want to collect, you can.
Yes. I also know, just from experience.
Yeah. Many other.
So there's a question about glass sponges. So glass sponges are deep species. So they can be very deep. I'm not going to say that how deep. We're talking about thousands of meters. And I don't know how shallow they can go, but I guess from my limited experience looking at these dives, we stopped seeing them at a few hundred meters. I could be wrong. Merrick Ekins is a glass sponge expert at the Queensland Museum, and he's a part of this expedition as well, although he's not on the ship. And later on in the trip, he's going to talk with us on the live stream, and he could explain to us all about glass sponges and why we're collecting them, and all questions about how deep they can grow, and how many species are described or undescribed. For example, when I was on the RV Investigator in the Coral Sea, we were dredging rocks, and we found little sponges on the rocks, and these are carnivorous sponges. They have little barbs that they use to pierce their prey, and they're like a centimeter in height, really, really small. We were lucky we saw them. And so, essentially, any rock you pick up could have some undescribed species on it, which is really exciting. And that's why we do what we do. Okay, we're now at 160 meters. It's 22 degrees Celsius, so there's now a distinct change in the depth, the temperature, and what we're seeing. So we're now seeing coralline encrusting algae. That's that green stuff you're seeing on the rocks. Ooh, can we stop here for a second? All right, so right in front of us, we have lots of little different corals, and I'm just going to have us settle down in front of this rock. We could just take a look at what each thing is.
Now you have time to do what you're doing.
Okay, I'm just going to quickly look at this coral in front of us. Okay, it looks like a soft coral. Can I get that autofocus off? Off, yeah, thanks.
Go ahead.
Thanks.
You want it that bright or you want to dim it a bit?
That's okay, that bright. Other way. Okay. All right, so yeah, looks like a soft coral. Why don't we grab a sample of this pink coral in front of us? I'll just get the lasers on it for a second to get an idea of the scale.
See them right there?
Okay, cool.
How much of it do you know?
Okay, that's good enough. So just a fragment of the side, any piece that you can grab, like here. So a lot of these corals are filter feeders. So they have polyps with tentacles, and the polyps are essentially the mouths of the animals. And so what they'll do is, with those tentacles, they'll grab moving particles out of the water and push them in their mouths. And that's how they survive. So we could put it in the back box 3-4, I guess, unless it fits in 2.
4x0. $5
I'm not sure. It could sometimes it indicates new growth.
Good for shipping.
Yeah, actually, so now that we grabbed it, it seems a lot harder than we thought, which is cool. Honestly, not sure what it is, so we're gonna sample it, of course, and figure out what it is back in the lab. Yeah, I thought it was a soft coral until we grabbed it with the metallic arms, and it seems to be very hard. Sorry, we're ending our dive, so we just have to get this in the bio box, and then we're just going to, I wish we could stay here at this rock and sample for another hour, but that'll have to be for the next dive. So we're gonna just observe.
Do you need a rock?
I don't think we'll have time. So if we can just keep filming for as long as we can until we have to come up.
Yeah, so we'll just head towards the shallow as we can. Thanks, guys.
You can definitely do a quick snag and grab on a rock.
You ready?
On the fly.
Oh, yeah. Jerry, did you see
You can do a real quick one, but
130, right?
Yeah, I think you're right, Rob.
We'll get it for you, Marty.
Yeah, Stalosterid, hard coral. I really thought it was a soft coral before we grabbed it.
I gotta come up 20 meters.
Bleaching can occur in deep water, so there's no reason why it can't. It's just less likely for it to happen. But if an event is extreme enough, or if the duration of the event is long enough, it'll have an impact. But generally, you know, effects of global warming are seen in the top, 50 meters. I think that's where it's being studied most closely, so that's why we know more about it. Yeah, thanks, Rob. So the tips, the white tips of the coral are not indications of bleaching. It can also be an indication of new growth among some corals, where when they first grow, they seem to be white or light colored before they fill up with their symbiotic algae. We have not yet come across any wrecks during these dives, but you never know. It's always a possibility. Huh?
Too soon? Too soon?
Nearly a wreck.
130.
Keep your eye out for a spot where you can settle and check grab.
There's one right there. Oh, yeah. Feels overhand?
Yep.
Where?
Yep, thanks, Rob. So the 2016 bleaching event, Tom Bridge surveyed corals for bleaching, and down to 40 meters found effects from bleaching out in the Coral Sea. Sea blankets to protect corals from sun. It's still not mitigating the primary drivers of climate change.
Go ahead. Yep.
Too small though?
No, I'll take it.
Put it in what, D?
C, 1C.
Roger. Watch out.
That was pretty easy. Can we try for one more?
Of course.
Yep. Same spot is fine. Yeah.
Tom, you can see thermoclines on dives. Do you often see thermoclines? Yeah.
Yeah, we can catch them in our fork day.
Yeah. Yeah, we are certainly cutting across, from almost 0 degrees Celsius up to warm, 30 degrees Celsius. So, yeah, thermoclines can be observed. Anything anthropogenic? Is that what you mean? We haven't seen any human impacts on this dive, but if you were watching or want to look back at yesterday's dive, we went to Cairns Seamount, and it's a popular fishing site, and therefore we saw lots of discarded fishing lines, and we saw a can, like a Coke can. So there were definitely there's evidence of human. There's human debris on the Cairns Seamount, but it's a popular site, so you know there's people there a lot, and
I'm not positive, Marty.
it's not surprising.
I think that was the base.
That one right there, right?
Yeah.
Sure. See it, John?
Oh, shark. You guys can't see it, but there's a shark behind us.
They saw the very end of the day.
Oh, they did?
Yeah, I had to
Nice.
wake up the tablet to get the chicken shell.
Same spot, Marty?
Same spot, thank you.
That's enough?
Yep. You can go and see things. Much appreciated, thank you.
You're welcome.
Okay, guys, we're going to keep the stream going for now, but this is the end of the dive. So we're going to head a little bit shallower before we bring the ROV back up to the surface. So at this stage, I just want to say thank you all for joining us on this adventure. We saw a lot of really amazing things, most notably the black coral on the Nautilus shell, but could be biased. So yeah, thank you guys so much for tuning in, and please keep an eye on dives in the upcoming days as we explore more seamounts, reefs, and cays in the Coral Sea and the GBR.
Okay, Javon, just for information, the cape is turning towards the port side of the vessel.
Yep, Roger. We know we're moving the vessel forward right now. Thank you. Sure.
Okay, just get back.
Yeah, and that. It's like the hammer drag. He was going that way. What it's happened.